Good afternoon, everyone. My name is Tess Taylor, and I'm a Community Education Specialist at MDA. We're glad to have you join us today for our ALS Virtual Learning Series. This learning event, which is a two-day event, is part of our larger MDA Community Education Program, which focuses on bringing the neuromuscular disease community together around education and resources. Be sure to visit the Community Education section on mda.org for updates on upcoming events. We are recording today's event and we'll be posting it to the website for on-demand viewing at a later date. Please know that all phone lines have been muted, but you can still ask questions at any point. Simply use the chat feature to type in your questions and send them in as you think of them. We'll have a Q&A session at the end of each presentation. MDA would like to thank Mitsubishi Tanabe Pharma America for supporting today's virtual learning series. For nearly 75 years, MDA has led the way in funding research to accelerate the development of new treatments, expanding access to care and clinical trials through the MDA Care Center Network, and advocating for access and inclusion for the neuromuscular disease community. MDA delivers its mission through research, care, advocacy, and engagement. Since its inception, MDA has invested more than $1.1 billion in neuromuscular disease research. MDA funds projects through the drug development process, from early discovery through translational stages and infrastructure support for clinical trials. MDA care centers are located at over 150 top medical institutions nationwide, providing care from clinicians who specialize in neuromuscular disease. MDA's advocacy work focuses on support for accelerated approvals and novel research, access to healthcare, treatments, independent living, newborn screening, accessible air travel, and more. The focus plays a vital role in shaping laws and policies that support the needs of people living with rare neuromuscular diseases. Our community education and recreation programs are designed to connect individuals and families. We are committed to building a nurturing environment where our members can share experiences, triumphs, and challenges. In 2023, we offered over 40 educational programs, over 20 summer camps, and dozens of other community activities, both in person and virtually. If you haven't done so already, we invite you to join the MDA community by registering with us. Registration is free. Please see the link in the chat. To learn more about MDA, please visit mda.org. We'd also like to recognize that May is ALS Awareness Month. MDA is dedicated to supporting the ALS community. Over the last 7 decades, MDA has invested $176 million in ALS research. We are currently supporting 28 research grants, and our nationwide network of care centers has 47 ALS-specific centers providing multidisciplinary care from specialized clinicians. We invite you to check out our community education resources for ALS listed on this page. They include the at-home physical and occupational therapy exercise guide that will be presented during tomorrow's session on ALS and exercise. Please know that MDA is with you in this fight. To learn more about MDA's efforts in ALS, please visit mda.org/ALS with MDA. MDA is excited to bring you this great lineup of presenters over the next few days who are here to give us up-to-date information on ALS. First, we will hear from Dr. Lyle Ostrow regarding advances in research and care for ALS. After the opening presentation, MDA's Vice President of Public Policy and Advocacy, Paul Melmeyer, will share some important updates related to the ACT for ALS. Later, Sarah Dramstad will be here to discuss nutritional considerations in ALS. Looking ahead to tomorrow, we will have two more presentations and 3 presenters. Kristen Larson will be here to discuss speech and communication with ALS. And we will round out our series with Angela Escalante and Deborah Taylor, who are here to discuss exercises for daily living with ALS. We encourage you to attend both days of presentations so that you can receive this great information and ask questions from our presenters. Now it is my pleasure to introduce our first speaker, Dr. Lyle Ostrow, MD, PhD. Dr. Ostrow is a neurologist and ALS researcher at the Lewis Katz School of Medicine at Temple University. He is a member of both the Scientific Advisory Board for Everything ALS and the Board of Directors of the ALS Hope Foundation. He is director of the Temple University ALS Postmortem Corps, which is funded by a collaboration with the CDC National ALS Registry. He is also director of the Temple Nerve and Muscle Biopsy Service and Neuromuscular Pathology Lab. Dr. Ostrow is chair of the programmatic panel for the Department of Defense ALS Research Program. Presently, the largest dedicated annual funder of ALS therapeutic discovery and validation. He serves on several ALS steering committees, advisory boards, and review panels, and has given over 40 invited talks, grand rounds, and keynote lectures on ALS research, biomarkers, postmortem tissue banking, and harmonizing multi-center research efforts and core resources. Dr. Ostrow, thank you so much for being here today. I'll turn the time over to you.
Thank you so much. Hold on, let me see if I can share my screen. You can see my screen?
Yes, we are good to go.
Okay, and you can hear me?
We sure can.
Okay, so thank you guys so much. I'm really just grateful for the chance to talk about this stuff. And I talk fast for people who know me. I'm gonna talk about a bunch of stuff. I'm gonna pop through some slides pretty quickly, but my understanding is that you guys are going to record all of this, and so it will all be available. I do want to point out, so I do have, I have research support from the NIH. I have a grant with collaborators at University of Texas and University of Virginia, looking at a particular ALS therapy development program. And as you mentioned, from the CDC, ALS Biorepository, which supports our postmortem efforts, and really we wouldn't be able to do what we're doing without their support. My roles with the Department of Defense ALS Research Program and with the ALS Hope Foundation and with Everything ALS, these are volunteer jobs, and I'm grateful and really just love the opportunity to contribute to these programs, but I'm not funded by them, I'm not employed by them. I want to very specifically say that I don't speak for these programs. I specifically do not speak for the DoD or the CDC, because I'm going to tell you guys some things that are my opinions and what I'm hopeful for in the future. And in a very personal way. So with that, I also want to start by saying, where do I get my information? And this is something that's changed over the past year or two. And it's just remarkable how much is happening in ALS, right? And how it feels like if you don't check your email for a day or social media, that you're going to miss something. And the plain fact is, the way that I keep up on everything that's going on in the science and in clinical care and in trials is largely due to people living with the disease and resources that are community-built, resources. And these are just a few examples. I'm not trying to say that this is all that's out there, but these are some of the places where I personally get my information, and I would very much encourage people who are interested in staying up to date on stuff in ALS to check them out. And certainly the MDA website has a lot of great information on research and on clinical care. There's some unbelievable clinical trial navigators to search for information on trials. This is one of them at ALS TDI. There's one at IMALS. There's a few others there that, these were built by people living with the disease, and they're remarkable tools. And right now we have this growing interest and growing community of people who have genes that we know cause ALS, but they don't have ALS. They're at risk because they have a family member has ALS. And, we have to figure out how to follow those people in clinic and how to do trials on them. And really, and the legacy, I think, is leading the way in a lot of that sort of thing. I wanted to just quickly, and I share this thing up here that probably nobody's heard of, and is like, what is that? Metals. And I made this little URL myself, and I actually there's no way to share this on a slide. And so this is something that somebody sent to me that I need to call out because it's amazing. And it was made by a person living with the disease, and I don't know them. And I just this is a resource. If you are on this call, if you guys know this person, just please thank them for me. I'm incredibly grateful. This is not a scientist, but a person living with the disease. And they update this once a week. And it is the most unbelievable source of every single drug development program in ALS, and basic science through to clinical trials and everything about them. And so there's a list of meds, right? Every med that's in development, and the publications and the links to the companies and what's known about them. And it's the kind of thing that is just a remarkable resource. Now, I don't endorse, there's some, subjective opinions on here, but as a source of information and references in this, data heavy interconnected world we live in, it's just remarkable. And so I encourage you guys to check this out. It's got so much information that it takes a few minutes to actually load. I don't know who this person is, but this is an example of how our community is really just, doing remarkable things. And so with that, in this world we live in where everything's going on at once, Terry Hyman Patterson, Dr. Hyman Patterson, who's my partner in the clinic at Temple, she's giving a talk right now too, and right at the same time. So there's a lot going on in this ALS community, and I put this on here because she's one of the people that I admire most and strive to emulate, and I'm sure she's giving an awesome talk right now. And so check it out. I'm guessing that it's going to be recorded also, and something that's going to be well worth listening to. So MDA asked me to give a talk similar to this about a year ago. I think it was one of the first kind of in-person things after COVID. In this session, this updates in ALS research, and it was chaired by Dr. Harms, who's in this picture over here, who's the MDA's medical advisor. And so when I was asked to give this talk, I kind of wanted to see like what's changed. Let me look back and see what I was talking about then, and what maybe has changed in the past year, and what really is different. And it's quite remarkable. It's quite remarkable how much has changed. And so when I gave that talk, I started off with a slide talking about Dr. Griffin. And Dr. Griffin was one of my mentors. He's passed away. He was the chair of neurology at Johns Hopkins when I was a resident and a fellow, and then joined the faculty. And he also was the editor of Nature Clinical Practice Neurology. And when I gave that talk a year ago, I started off talking about this editorial that he wrote, right? And research in neurologic diseases lost in translation. And the point of this article was that we were learning so much about the biology of these, neurodegenerative diseases like ALS, but it wasn't translating into effective therapies. And there, this was written in 2006, but it's the kind of thing that could easily be written today, right? And in Dr. Griffin's essay, he talked about what his thoughts were on what we had to do to fix this. And this included focusing on speed, being able to, fund and research quickly, and having flexible funds that can be directed promptly, requiring unfettered exchange of ideas and data, truly being able to share data broadly and share information, and then having, approaches to doing therapy where we're not being delayed by somebody worrying about authorship or ownership or intellectual property. And you know what? This stuff is happening now. And so why do we have a problem, though, in general, historically translating therapies in ALS? And so this is the rub, right? So we do all this stuff in the lab, in mice and cells, and we have trouble translating that into successful therapies. And so we talk about, is the problem that the models that we're using, the cells or the specific animals that have a genetic mutation that causes a kind of ALS, maybe they're not a good model for everybody with the disease, maybe only certain people with the disease. Or maybe we're not testing these treatments in the right time. And so we think about, how can we do a better job of validating which people living with the disease are the ones that we should be enrolling in which trials? And how can we tell if the drugs are doing things to the biology in people's bodies that they were doing in the lab? What can we measure to tell if a treatment is working? This is a more recent, this is a review that's open data. I encourage you guys to go check it out. It's really a nice review. ALS, a neurodegenerative disorder, poised for successful translation. So why? What's changed, and what makes it that we can maybe have success now? And these are some of the points that are talked about in this article, right? That we've learned a lot more about the genetics and the biology, and we're going to talk about some of this. That we have biomarkers, right? Ways to tell when somebody is becoming symptomatic, and ways to tell if our drugs are engaging their target in the cells and doing what they're supposed to do. Specific pathways that we can target with therapies, genetic gene therapies, which, a few years ago were still science fiction. And it's really a good review. I'll tell you, though, it misses the boat on one thing. And the one thing that I really think it misses the boat on is the role that people living with the disease and their family members and advocates have played in every stage, every aspect of therapy development for this disease, and really in an impactful way in the past few years. And so I want to highlight some of those things too. So to talk about this, these are the things that we talk about, right? So what is ALS? What do we mean when we say ALS? How do we search for therapeutic targets? How do we decide that something is a pathway that might be important? What does it mean when we say validate that? We got to validate this. What does that actually mean, right? Why are some things in trials and not other things? If I go on social media right now, I'll find a bunch of posts saying, how come nobody's testing this? Why is this not being tested? Why aren't we doing this, right? So why? How does that happen? And, why have some trials historically not been successful in ALS? And a common refrain we hear again, like, what's the difference between why can't we do what they do in cancer or in HIV? How can we de-risk industry investments so that a company doesn't have to say, oh, I don't want to invest in ALS because the trials fail. Why would I want to do that? Like, what can we do? What do we mean when we say biomarker? What are the different kinds? What do we need? And how can we harness this interconnected world and this data-heavy world we live in to really do this better and do it more efficiently? So, and by the way, I have a hard time giving these talks nowadays because the audience is so much more educated, right? You guys know so much and you understand this stuff. And it's sort of like, I feel as if I'm talking to people who already are aware of these things, but I do think it's important to talk a little bit about what do we mean when we say ALS and why is that relevant, right? So, ALS is a disease of progressive weakness, right? It can start anywhere in your body. It can start in your thumb, it can start in your breathing muscles, it can start multiple places in your body, and then it spreads. And ALS is a fatal disease because eventually it involves the breathing muscles or the swallowing muscles. Some people with ALS also get a kind of dementia called frontotemporal dementia. That's more common with certain mutations, but it's still a clinical diagnosis. We're pretty good at diagnosing it. We're not good at getting people to an ALS clinic to be diagnosed, but we're pretty good at recognizing ALS. But it is a clinical diagnosis. Aside from the few genetic types of ALS, which are just a small proportion, we don't have a test to confirm it. And this is a big difference from, say, trials in cancer. If I'm developing a drug for a kind of tumor, well, I can see, did the tumor grow? Did the tumor shrink? Did the tumor stay the same size, right? If I'm developing a therapy for blood pressure or diabetes, I can see what happened to the blood sugars, what happened to the to somebody's blood pressure with this medicine. And in ALS, it's more challenging because we rely on clinical measures. And every person living with the disease is different. In some people, it's faster. In some people, it's slower. In some people, it involves different parts of the body. So, those are the challenges that we have to overcome. And so, 90% of cases of ALS are ones where we don't have a family history, right? We say sporadic ALS. 10% have a family history. There's a small portion of the ones that don't have a family history that we still find gene abnormalities, but we keep finding more genes that are involved in this disease. And so what this plot is showing is the x-axis here, these are years, right? So SOD1 was the first gene that was discovered that causes ALS back in the early 1990s. And as time has gone on, we've discovered many, many more genes. And that also correlates with us learning how to do genetic analysis in more efficient ways that let us detect these things. And so this is from this paper from 2015. Oh, and the sizes of these circles are how common they are. So the most common genetic mutation is this C9orf mutation, right? Second most common is still that SOD. And this is a figure, this is an updated version of that figure that was made for me by Dr. Harms, who's the MDA medical advisor. And it's showing that, again, as we've gotten even these newer next generation techniques, as the years have gone on, we keep finding more and more genes. And some of these genes appear to cause the disease. Some of these genes appear to increase the risk of the disease or change the way disease progresses, but we keep finding more genes. It's interesting. This first gene, SOD1, was discovered back in the early 90s. And right around that same time, we had our first animal model, right? So this was published in 1994. This is a mouse that has a mutation in that gene, this SOD1 gene. And they develop a disease that looks kind of like some kinds of human ALS, right? And so in the lab, we can test therapies in this mouse that has this mutation. So, we discover our first gene, they have an animal model. That's right around the time that our first drug was approved too, Riazol. And interestingly enough, Riazol wasn't tested in this mouse model. But Riazol slows the disease down. And we now know that it slows the disease down considerably more than we thought it did, especially if given perhaps earlier in the course of the disease. But every trial that's been done since Riazol came out of a new therapy, there's people who are on drug and people who are on placebo. And a portion of the people who are on the placebo also happen to be taking Ryuzol, because you're allowed to take Ryuzol in these trials. And so over and over, it's been confirmed that it slows the disease down. But you have to think, back in the early 90s, we discovered a gene, we had our mouse, we had our therapy. Everybody must have thought, wow, we're going to have this disease beat quickly. And then there was a long period of time where we've learned a lot about the biology, but not until recently, in the past year or two, have we started to really get signals again and ways to treat people and new therapies. So this is a figure from that same review that I just showed you. And when I was in medical school, we had this thing we called that when we were sitting in lectures, like we called them OHEC slides, right? We didn't use the word heck, but you get the picture. And so this would be an OHEC slide, like way too much information on a slide, right? And so if a lecturer showed me 5 OHEC slides, that's when I'd say, you know what, I'm not paying attention anymore, and I'd like doodle or do something else. And so I want to be clear. I'm not showing you this to say, hey, look at all this information. I'm showing you this to say, look at all of these different mechanisms and all of these different genes, and to make the point that they impact different parts of biology, right? So this is a picture of a nerve cell. Some of these impact other cells, microglia, astrocytes, other cells in the body. But all these different genes, one of the things that we still don't understand, and that it's really important, is how is it that, none of these, this is not like your ALS genes, right? These are genes for proteins that do important things in every cell in your body. And somehow this diverse list of genes can cause this very specific disease, ALS, right? And some of these genes are implicated in other diseases too. Sometimes, things like dementia, but also other diseases in the body that have nothing to do with the brain or the nervous system. And so how does that work? And that's one of the puzzles that we still haven't solved. It's also interesting to note that a lot of the mechanisms that are impacted by these genes are very general cell mechanisms. We all learned in high school, right? Mitochondria are the powerhouse of the cell, okay? But how is it that I can have these mutations that affect mitochondria and only get this specific disease? And even in the people who don't have a genetic mutation, we have this growing list of mechanisms, of things that are going wrong in the biology. And it makes my head explode a little bit. I guess that's supposed to be me in the corner there, right? And so, again, these are often very general cell mechanisms. But where does this list come from? And we're going to talk about that a little bit. And why do we think that these pathways are important? And what we've learned as we've gone on is not only are they important, but every person living with the disease has there's different clusters. So in some people, certain mechanisms seem to be the bigger problem. Again, from that same paper, another OHEC slide. I'm on 2. You guys can, don't try to read the slide. But this is making the point again that look at how exciting this is. So these are all different clinical trials targeting these different mechanisms. There are a ton of trials ongoing right now, and this isn't even updated. And again, if I want to know an updated thing, I go to the ALS TDI or IMA ALS, or these unbelievable resources that are being maintained by people living with the disease. But there's an unbelievable number of trials right now attacking these different mechanisms for people living with ALS. Now, the problem is we still don't always do a good job of figuring out which people should get which drug, right? If you're in a therapy, in a therapeutic trial for something that affects your mitochondria, boy, I'd like to know that you're one of my patients who maybe your mitochondria aren't working as well, right? Or if you're somebody who has more or less neuroinflammation. And so one of the kinds of biomarkers that there's a lot of work to develop right now are what we call predictive biomarkers. Things that I can measure in somebody, and it will help me predict, will they respond to a given therapy? Are they the ones that I should enroll in a particular trial? Because if we're enrolling everybody, maybe that's why a lot of these trials fail. As an analogy, if I have a trial for a genetic form of ALS, like this medicine that was just approved, tofersen, right, Qalsody, for SOD1 genetic ALS. If we had enrolled everybody with ALS, the trial would have failed. In order to get into the trial, you had to have a gene test that showed that you had a mutation in SOD1. And, of course, that makes sense, right? Nobody would question that. And so the gene test was the predictive therapy. We're not doing a good enough job of that across the board for these non-genetic therapies, but it's changing. We're starting to. So where does that big list come from? And what do we do in the lab? And how do we look for therapies? And it's really pretty simple. And so the idea is, whether it's in a mouse model of the disease or cells growing in a culture dish or whatever the test might be in the lab, you have your normal and you have your disease, right? You have two things that you're comparing. And you're looking for things that are different. And oftentimes it's, maybe it's obvious. Maybe they just really look different. The cells look different, like an apple and an orange. But sometimes the cells don't look different, right? Sometimes you can't really tell the difference between the normal and the diseased apple, okay? But if you grind them up and make applesauce, and then you analyze that applesauce, maybe you can tell that one has more sugar or one has a different kind of chemical. And this is legitimately how lab experiments often work, right? We're taking, tissues from people living with a disease or from animal models or cells, and maybe we're homogenizing them. We're making applesauce of those cells, those nerve cells, let's say, and then analyzing their proteins or what's inside them and looking for differences, and then looking whether we can, after we discover those differences, can we use a drug to change that? And one of the things we think about a lot, is, does it matter? Like, so if this is a tree with apples, right, and the tree is sick, and the apples are falling off the tree as the sickness progresses, does it matter if I look at the apples that are on the ground that have already fallen off the tree, or the ones that are left on the tree? And so, for example, if I'm doing an experiment on let's say, autopsy tissues from somebody who passed away from ALS, maybe the cells that are left at the end of the disease, the ones that are still on the tree, are there because something's protecting them. Maybe if there's something different about them, it's a good thing, not a bad thing. And so these are the sort of things that we think about. And things are even more complicated than that, because if I have cells, nerve cells, and I grind them up like apples, that's not really representative of what's going on in my body. My brain is more like apple pie, because there's apples and then there's other ingredients. And as the disease progresses, the ratio of those ingredients changes. Maybe you get more inflammatory cells. Maybe you get less apple because, the motor neurons have died. And so if you grind up the apple pie and analyze it, maybe you're going to get a different answer because the proportions have changed. A lot of times we look for markers, we look for ideas about things we can, track as the disease progresses in biofluids, in spinal fluid, right? Or in blood. And in some ways that's like this ice cream melting off this plate here, and we're measuring something in it as it's picking up some stuff, as it's going by the apple pie. And so we have to make sure that what we're measuring is relevant, and we have to make sure that what we're measuring is something that's important in people living with the disease. So this is an example, and this is it doesn't really matter what the particular drug is, but these are mice that were all litter mates. They all were born from the same litter, and they have that SOD1 mutation. So these are these SOD1 ALS mice, and the ones on the left here were given placebo, saline. The ones on the right were given this medicine. Okay, and if I play the video, I think you can appreciate that the ones on the right, they move around more, right? These guys aren't moving so much. But if you really drill down and look at it, there's one dude here who is moving better. And maybe you'll notice too, the mice are different colors and they're different sizes, and they don't look the same, right? And so one of the things that we've learned about these mouse models is that we have to be very careful about the variability in the models. And are we using the right number of mice? And does it matter if they're male or female mice? And so we've learned how to use these models better to do more rigorous statistics and do more research. And then, so this is a mouse with an SOD1 mutation. Maybe this therapy that's listed here, maybe it works in these mice, but maybe it's not going to work in a person that doesn't have an SOD1 mutation. And so we have to figure those things out. So this is my house. I live out in the woods in this old stone house off the grid. Like, we bought this house about 10 years ago, and it had been vacant for many years, and it was a disaster. It's the only reason we could afford it. Everything was broken, windows, appliances, mold. But we bought it and we fixed everything up. And then that first winter that we were living in this house was us. I live in northern Baltimore County, and we had a big snowstorm. And what we discovered was that our house, this old stone house that was built in the 1940s, was unbelievably drafty. And so if it was blowing wind outside, it was blowing wind inside. And our house was cold, and we had trouble staying warm those first many years. And so if you didn't know what made a house cold, and if you didn't understand why a house was cold, and you were doing an experiment and you said, let's compare this house, the disease house, to the control house next door, and see what's different. I'll tell you what, that first year, what you would have found is that maybe we had more ash in our fireplaces because we burned a whole lot of fires, right? Now, if you erroneously concluded from that, oh, you know what, we should get rid of the ash. That'll fix things. That'll make it better. Obviously, that wouldn't be true. Interestingly enough, a lot of the things we see under the microscope in ALS are not dissimilar to ash. They're proteins that maybe clump up in places they're not supposed to be. And so in the upper right here, this is showing this is a protein called TDP43, and it's normally in the nucleus of the cell. And in disease, it goes out into the cytoplasm and it gets trapped there and clumps up. And, you know, we have to think, is this ash in the fireplace or is it doing something bad? In the case of TDP43, we know it's not just ash, it's doing something bad. And in fact, we often talk with genetic mutations or with mechanisms in ALS, like, is the fact that it's getting stuck out there, is this clumpy stuff doing something bad, what we would call a gain of function, or, you know, maybe blocking something else out there? Or is it that it's not able to do what it's supposed to do in the nucleus, what we call a loss of function? And sometimes it's not so simple, sometimes it's both. Now, if you understood how fireplaces worked and you said, yeah, but, you know, you were burning fires because you wanted to make more heat, and you know, and so you need to burn more fires. We actually tried that, right? But what we learned is that burning more fires actually made our house colder because it pulls in drafts, right? Because everything in our cells and everything in my house, everything's connected, okay? So this is our furnace, and I'd never had, this is an oil furnace. This is outside our house. This is the oil tank. That's a stopper that's supposed to be up here showing that we've got oil. This is the middle of the night because we ran out of oil, right? Because we burn much more oil than my neighbors. If you compared our house to my neighbor's house, we burn tons more oil. Again, you don't want to conclude that the trick is to burn less oil. This is our furnace. It breaks down, I guarantee you, more frequently than my neighbor's furnace. But maybe that's not because there's something wrong with it. Maybe it's because it's working harder. And so a lot of the mechanisms that we you know, identify in cells that are abnormal, you know, these are all they're mechanisms that have to do with energy use and transport and, you know, ways to for the cell to respond to diverse insults. And so we often have to wonder, are these possibly, you know, the furnace breaking down because it's working too hard, or you know, using too much oil, or are they a specific problem? So what's the point? The point is that, you know, again, ALS is a clinical diagnosis. We know that there's a lot of different biological causes, and that we also know that in people living with the disease, that it's the product of multiple interrelated factors. And what we've discovered from looking at these large sets of samples from people living with disease, whether it's, you know, cells that we've taken from our patients and turned them into stem cells and analyzed, and then into nerve cells and analyzed them, or tissues after somebody passes away, or blood or spinal fluid, that there's subsets, and the different subsets of people living with the disease seem to have different dominant mechanisms that are abnormal. Now, none of that would be possible to do without the unbelievable donations of these resources of people living with disease, right? We couldn't even do this research. But we know this, and so it's sort of foolish to think that a therapy that targets one mechanism is going to work for everyone, and that's where we really need predictive biomarkers to tell us, you know, which people should get which therapies. And we still don't understand how all these different mechanisms and diverse mutations all cause this very specific disease, ALS, right? That's important too. Now, in my house, the genetic mutation, the main problem is that we've got these steel sash windows that I can't replace because my house is made of stone and it's 2 foot thick. And, you know, I would tell you that genetic therapy, you know, gene therapy is also hard, right? But it's becoming easier, and it's becoming easier quickly. You know, people with SOD1 mutations now get tofersen, and they're showing some improvements. People with other genetic mutations, there have been people treated with a rare mutation called FUS. They're you know, a different kind of genetic ALS. And I encourage you to search the internet and find the trials, some done by Dr. Schneider at Columbia, but these people with a targeted therapy against these gene mutations are improving, right? And so gene therapy is a real thing, and we're getting better at it quickly. Now, this is my my father-in-law. He's passed away, but they used to come visit us every Christmas you know, and he would complain about how cold our house was, and he would turn the heat up in every room, like, and we would follow him around and turn the heat down because we were burning $80 of oil every day. And so for Christmas one year, we bought him these footy pajamas, right? And so this is a good treatment. This isn't affecting my drafty windows. This isn't actually affecting the gene mutation in my house, but this is making it so he doesn't feel cold. And, you know, it had this awesome side effect too of this treatment, that he looked hilarious, which the rest of us appreciated. But the point is that effective therapy doesn't necessarily have to target the primary, you know, cause, the mutation. And those mice that I showed you that were treated with that drug MG53, that's one study that I'm involved in. And by the way, you know, I'm using some examples today. I'm not saying that these are better or worse than other therapies. Any ALS clinician or scientist who tells you one trial is better than another, one therapy is more promising than another, are absolutely full of baloney. If we knew, we would have done a better job in the past 35 years of having successful trials. What we know is how to do rigorous science and hopefully how to validate these things. And we're starting to see some improvements in, signals in trials. But if somebody tells you that one therapy is the one to be excited about, don't buy it. Anyway, what that drug does, that MG53, is that it actually improves the ability of muscle cells to repair their connection to nerve cells, right? So it doesn't actually target the cause of why my house is cold at all. It's like the footy pajamas. It's making it so that your muscle cells can repair more quickly and stay connected to nerve cells. This is some of the data from that paper, and I just wanted to show you because it's the kind of data. So one of the things that's really important is that you guys have access to all the science now, right? And we all need to be responsible consumers of data, and we all need to understand what we're looking at. And so this is sort of typical experiments that are done with mice with a mutation. And so this bottom corner here is showing there's 100% of the mice are surviving at the beginning, and over time, as mice die over different days, these curves go down. And the red line here is the mice who got placebo, and then the black line is showing that the mice lived longer with the drug, right? These things on the left are interesting. So, we don't bring the mice into our clinic and tap on their reflexes and check their strength, right? They're living their life happily in their cages. And we have these movement tracking software that are videoing them and automatically keeping track of their movement. And seeing that the ones that got the drug are moving around a lot more, like in that video I showed you. And we're going to get back to this because, I don't need to be there watching these mice move and timing them. This is an automated thing. And it's sort of analogous to a lot of the work going on right now that we're able to do with digital biomarkers in people living with the disease. So, where do those studies come from? How do they get funded? And I always like to point out, so the reason that the US has led the world in this sort of research for decades was because we were the first country that had this idea of peer review, that NIH formed these things called study sections, okay? And the idea was that the grants weren't going to be reviewed by a good old boys network, or people who weren't scientists. They were reviewed by peers who had expertise and were tasked with, reviewing all the science. And it really is what helped our country, I think, lead for many, many decades, in basic science research. By the way, the first study section was for treatments of syphilis, okay? And we'll get back to that. But, unfortunately, the way we've done this hasn't changed so much in 75 years, and it takes too long, right? And this is a slide, I'm not going to read it to you, but the idea is that, a funding agency, whether it's governmental or a foundation, makes a request for proposals, and then people send, sometimes pre-proposals first, and those get reviewed, and then they get invited to send a full proposal. And this is a time-consuming and effort-consuming thing. We're finding ways to speed it up, but generally this is the way that this still works. And here's the problem, right? So how do those reviewers choose? Because we don't have money to do everything, right? When you say, why aren't we doing this? Why aren't we doing this? It's because all of these proposals, only a couple can get funded for most mechanisms. And so, we need to make judgments about what's the impact. Are the experiments well designed? What we hate more than anything is getting to the end of a research grant and not having an answer. Like, it's fine with me if the answer is our hypothesis was wrong, but not having an answer is not acceptable, right? And so, these are the kind of things that peer reviewers and committees consider when they're reviewing grants. And in ALS, it's particularly challenging because until we have, a bunch of therapies that work, it's hard to say which of these models are the ones that are going to be most meaningful and translate. This is that first study section on syphilis for the NIH. And I wanted to show this picture for a couple reasons. So, one is they don't have this interconnected world that we live in, all this data. They've got their piles of paper. Another is, this is not a diverse group, okay? I mean, the only diversity in this room is that some of them have different colored suits. Certainly there's no people with lived experience of the disease, but really it's all a bunch of, I guess some of them have glasses too. Other than that, there's no diversity. And so we've come a long way since this. And in ALS, so this is a slide that I actually borrowed from the DoD quite a time ago. This is the way that we have organizations dedicated to different aspects of therapy development. And so, early epidemiology and looking at risks and cause factors is the purview of the CDC, the ALS registry. basic science. And by the way, this is only listing examples of organizations, certainly MDA and many foundations and many other organizations fund along this spectrum here. The biggest funder, though, the early biology and mechanistic is probably, still NIH. The DoD program, which I'm a volunteer for, is firmly planted here in this immediate preclinical development, therapy development. And now we've been able to move into early phase trials as this program has grown and trying to get things to translate from the lab to the clinic. And I also want to point out that late stage trials in ALS are still funded by industry. And I think that this is suboptimal, and maybe we'll talk about why in a bit. And maybe someday we'll be able to fund those not entirely by industry. But here's the thing, right? So every time I talk about this stuff, every aspect of this pathway has been massively impacted in the past couple of years by people living with the disease, whether it's listening sessions and helping to improve the registry and their national meeting and what they're focusing on, increasing funding, increasing the design of clinical trials and making trials more equitable and data efforts more equitable. Every single aspect of this pathway has dramatically improved, and it's because of advocates and people with lived experience. And so the DoD, the ALS research program that I'm involved with, this is something that I'm most proud of. So this is, Sandy Morris, who's passed away, but started this idea, nothing about us without us, right? And we've learned that this really is, nothing could be more true. I started off by showing you how I rely on all these resources that come from people living with the disease. And we also rely on their input, your input in the research, right? And so the Morris Principles gave an award to the ALS research program, and it was because exactly what it says here. People living with ALS are involved in all advisory bodies, everything that we do, everything related to what is the vision, what should be our priorities, what should get funded, and that their scores of reviewers carry equal weight to that of the scientific reviewers. And this is the way it should be, and we should never go back. This is the budget from that program, from the ALS research program. And I joined it here in 2012. And the budget was around, 5 to 7 million. We could only fund 3 studies. We probably got 80 applications that year. And I got to tell you, no matter how good of a job we do, it didn't feel very impactful. It didn't feel like it was that useful because of the assistance of people living with the disease and industry and scientists all working together to improve these mechanisms, to make sure that we're requiring rigorous science and biomarker development and getting the actual funding mechanisms to help dictate what the researchers are doing and being responsible. Things that we funded have moved forward in the pathway, have moved from the lab to the clinic. And because we can demonstrate that that's working, the budget and because of advocacy of people living with the disease, the budget has increased dramatically. And so these numbers are the numbers of grants. Look at this, right? So compared to back then, 40 grants in 2021, 41. This is over 100 grants in 3 years, 100 new therapy development programs in ALS. It's just remarkable, and it's because of you guys. And and the start of this was all in FY20 when we had this what we thought was an amazing increase at the time, and it was, and it let us design new mechanisms to focus on biomarkers, to do things that we hadn't done previously. And it's really making a difference. If you go on the website, and here's the link right now, these are copy pasted from some resources that are there. These are just some examples of clinical trials of right now therapies that are moving through clinical trials that, that were supported early on by this program. And it's remarkable to see. And I wanted to really stress this because right now, these funding award mechanisms are open. And so please, please, please tell researchers tell your clinics, tell foundations. Industry can apply for these awards, nonprofit foundations can apply for these awards, clinical teams can apply for these awards. You don't have to be in the US, you can be anywhere. The only way that we can fund the best science is if we get the best applications. When I joined the program, we only had these 2 awards, these therapeutic idea awards, which were, kind of new ideas, no preliminary data, innovation, high risk, high reward. And these awards designed to do what's called IND enabling studies to get a drug to a trial, to fund toxicity studies and manufacturing and all the stuff that's needed. Because of this increased budget, we now have these other awards. So we can fund early phase clinical trials that include rigorous biomarker development, mechanism specific biomarker development, ways to tell who should we enroll, ways to tell are we targeting the biology correctly in people so we can use these tools in the later phase trials, like the Healy trials, for example. And also, mechanisms specifically to find subsets of people living with the disease and to define new biomarkers. And, most importantly, by the way, one other thing to mention, we require data sharing, right? Not IP. We don't require a company that's doing a clinical trial to share their secrets. But if you're doing a biomarker development program or analyzing for clusters, you are required to share that data. The data that you produce, even from the Therapeutic Idea Awards, the model, whatever it might be, we score the data sharing statements. NIH, other governmental agencies, foundations require people to write a data sharing agent, but they don't always incorporate it into the review process, and we do because it's important. And the other thing that's really important, and we should never go back, and it needs to be the norm, is that all of the review of these mechanisms, their design, everything about them involves people living with the disease, community collaboration, and it matters. Now, I mentioned, FY20 was the first time this budget went up. And at that point, we didn't have enough funds still to fund clinical trials. Clinical trials are expensive. We do fund early phase trials now. But and I also wanted to point out, so now this is our budget, right? So the Department of Defense has different disease funding programs. This is us, the ALS program, but there's a bunch of different diseases. And, we now have a budget that lets us fund these early phase clinical trials. We still can't fund later phase trials, the ones that are, like phase 3 trials. They're expensive. The programs in the DoD that have successfully translated drugs into these late phase trials and have those mechanisms to do so are these bigger ones, like the prostate cancer, breast cancer. By the way, and again, I'm not speaking for the DoD. This is my own personal feeling, right? That, if someday the budget was to increase, what I hope is that we would find a way to be able to fund later phase trials if the budget was increased. But that's just my opinion, not speaking for them. But why is it important? And I want to use this example. So there's this Alzheimer's drug, donepezil, Aricept, right? And it's an approved medicine. And it's often used as a case study for what happens when all of the late phase trials are funded by industry. And by the way, this isn't industry doing something evil. It's not their fault. It's just an implicit, but I mean, they're going to perhaps be more excited about results that are less impactful. In the case of donepezil, there were a lot of large trials. I believe it's 2 of them that were not funded by industry. The rest were. If you look at the actual data, and that's what these papers talk about, the data, the results of those trials were virtually identical, but the way that they were talked about, the rhetoric, the way that the impact was discussed in papers and in editorials was quite different. And so, I think that it's not just a matter of how the results are interpreted. If we are able to fund late phase trials, we can make sure that those late phase trials have the biomarkers, right? That it's not a drug in search of a disease because a company has a drug and they say, oh, let's try it in ALS. That we can make sure that they are enrolling the people who are most likely to benefit, because we can make that a criteria for getting these awards. We can make it part of the review process. And so maybe someday we'll be able to do that in ALS too. But in FY20, when the budget first went up, these were some examples. We couldn't fund clinical trials, but we started this mechanism that let us fund biomarker development and to look at clusters of patients. And these were some of the things that were awarded. And it was adding on analysis of biomarkers to clinical trials. Because I'll tell you what, when the budget is crunched for a clinical trial, the first thing to fall off is biomarkers, right? That's the thing that if we need to cut money, that's what happens. And so this mechanism lets it be added back on. You can add biomarkers on to ongoing, otherwise funded trials. This FUS trial, this is that gene therapy that I was talking about that actually seems to be working quite remarkably. And again, this let the data be collected and analyzed to help figure out how it's working. Look at this. This is a study by Dr. Geronimo looking at can we tell things about progression of disease with a smartphone, looking at speech and swallowing. Because boy, like if we can do that, it's sort of like those mice that we were just monitoring in their cage, right? They don't have to come to clinic. You can monitor people wherever they live. They don't have to be near a brick and mortar ALS center. We can involve underserved populations. It's amazing that it's an equity thing that everyone in our society has a cell phone, but it's pretty much true. And then some of these projects looking at analyzing publicly available data. Again, we require this to be shared, at creating a new outcome measure. Dr. Fournier at Emory University, which is now the ROADS, which is being used in several clinical trials, even though this was only a few years ago. ALS Therapy Development Institute, analyzing samples from people. This is the sort of thing that's moving the science forward. So again, we have those mice, and we were able to do this. And guess what? We're doing that in people. And so one of the organizations that I work with is Everything ALS. Everything ALS does a lot of digital biomarker work. We have this Radcliffe study where we're using Bluetooth shoe soles, right? The Bluetooth shoe soles that aren't that different from those mice, okay? And literally tracking how people are doing. And if you go to their website right now, and there's other programs out there as well, such as the TDI program, but you can join these studies and participate wherever you live, whoever you are, and help us develop these biomarkers. And as an example, so this is one of the studies. This is the speech study. Right now, if I want to measure somebody's changes in speech in ALS, I've got this ALS FRSR speech question, which a lot of you are familiar with, and it's 1, 2, 3, 4, and it's just not granular enough, right? It doesn't give me enough information to tell if somebody's speech is changing. And so Everything ALS collected this unbelievable data set. By the way, this data is publicly and openly available. And then has this idea that we can use machine learning and artificial intelligence, which we hear about a lot, right, to analyze that data and find more granular measures. The problem is, what do you compare it to? How do you know that something that you're measuring with, or analyzing with artificial intelligence is clinically meaningful? And the analogy, and this is from Dr. Ernest Frankel at MIT, is if you were the person who invented the first thermometer and all you had to compare it to was putting your hand to your forehead and seeing if you had a fever, how do you know that the thermometer is measuring something? And so what Everything ALS did was harness the wisdom of the community, find from our clinics speech language pathologists who work with ALS patients, Dr. Green, who's an expert in speech and language, and sit down and figure out what are the different ways that we could look at clinically meaningful changes in somebody's speech, what's been published, what do we know. Usually this is the kind of thing, figuring out how to rate something as a severity scale that takes years. And in fact, this is one study that Dr. Green had published. Look at this, it took 15 years to collect this data and to analyze it. And we didn't want this, right? We need to be fast. And so what they did, what these the SLPs and Dr. Green did, was they found every mechanism that was out there. They designed a pilot study. The Everything ALS team, this is the data science, the data analysis team, and Swapno's the platform team, quickly made a platform really quickly. And I'm not going to go through the details of what happened, but tested out a whole bunch of different ways to measure changes in speech, a whole bunch of different speech tasks, reading a paragraph, reading a sentence, different length sentences. And the speech language pathologists, they made them a platform where they could analyze these things really quickly. And so the platform, they get played, these speech recordings, and the one that ended up falling out as the thing that was the most reliable and easiest and quickest was something called listener effort. And what listener effort is how hard is it, how much effort does it take to understand somebody when they're speaking to you, right? And it's just a simple scale from 0 to 100. And, there's a simple platform and the SLPs evaluate it. And listener effort, we know it's clinically meaningful because that's the purpose of speech, is to be understood. And so quickly this study was done. Those speech language pathologists were able to rate in a very quick time over 7,000 ratings, right? And what this picture on the bottom here is showing, this is that scale: 0, no effort to understand, 100, these are the ones that are hardest to understand. And normal people, I mean, if you speak really fast like me, there might be a little effort involved in understanding me, but certainly people living with the disease, there was this very granular spread. This is looking at the different reviewers, and their scores correlated really, really well. So the different people reviewing the data were coming up with the same scores. And to give you an idea of the power, these are 4 examples of people living with the disease over time. This is over a year. This is what we have now, the ALS FRSR, right? And so if you're a control, it stays stable. And if you're a control, the effort to understand you is 0. If you have ALS, so this is a person who their ALS FRSR, what we use now, was stable for over a third of a year, and then became declining. But if you look at this effort scale, it picked up the decline much earlier, many, many months earlier, and was much more steady through the course of the disease. And if I play these for you.
A clerk reads the zip code and punches 3 numbers on his keyboard. I tried to put my personality over as slowly as possible.
I think you can, appreciate that it's harder to understand this recording than it is this one, but that wasn't picked up by our current tools, and it was easily picked up and reliably picked up by this effort score. And so,
17.
Oops, sorry, hold on, sorry. Okay, so we have a manuscript. Now we've analyzed all this data, and I'm not going to go through this now, but there's 2 really interesting things. This is just part of the data. This bottom thing here is using these machine learning algorithms. So you train the machine learning algorithms on on that data, on the effort scale data for a certain period of time. And then you see, can it make clusters? Can it tell who's a fast progressor, a slow progressor? And that's what this is showing, that it can show, some people who aren't progressing, people who are progressing quickly, people who are progressing at different rates. This is interesting too. So obviously this listener effort scale can tell the difference between controls who don't have any effort and people. This is from people who have, living with ALS. If you look at people who have onset in their bulbar muscle, versus limb, there seemed to be a difference in the speed they were progressing. However, interestingly, if we took out the people who didn't have any involvement of speech, so people who just had limb involvement, and we only looked at people who had speech involvement, whether their ALS started in their speech or not, the rate is the same. So whether or not somebody had bulbar onset ALS, whether it was their first symptom or not, if they had speech involvement, it progressed the same. So these are the kind of tools that can now be used to look for a change in a clinical trial to see if a drug is impacting these changes. And this is the thing that's so amazing, is the timeline. So, we started thinking about doing that listener effort scale in November of 2022. 2 pilot studies done, the entire data collection was done and analysis, and now it's completed and the manuscript's getting submitted right now. And this kind of thing didn't used to happen in ALS or anywhere else. And it's because of this interconnected world we live in, because of data sharing, because of unbelievable people living with the disease who are contributing to these studies and doing so in an equitable way, because it doesn't matter where you live, it doesn't matter who you are, anybody can sign up for these things, and it's remarkable. And that Rhodes scale that I mentioned that was funded by the DoD, by Dr. Fournier at Emory, a new way to track progression of disease, that's been converted. There's this thing called ALS Motor, which is now a telehealth scale that was designed originally for physicians to do telemedicine to track disease progression. It's now been made into this self-reportable version and incorporated into the Everything ALS app, and is going to be rolled out again, as the kind of thing that can be done at home by anyone, and hopefully will be a meaningful way to track disease and not require somebody coming into a brick and mortar clinic. And this just shows the way these different components of our ecosystem, like the DoD funding, helped fund this project. And now this community-driven Everything ALS is helping carrying it forward. So this is the point though, right? So this was an editorial from a few weeks ago, and it was specifically about, how what's happened, I guess, with Relivrio, and we're still not sure, even though it was pulled from the market, it may work for some people with the disease, and we just need to figure out how to identify those people. But we have to stop treating ALS as one disease, and we have to stop thinking that, that these trials are going to be successful if we can't identify the mechanisms. And that's where why biological samples from people living with the disease or autopsy tissues are so important, right? This is how we validate, is what we see in the mice or the cells, is it, or, something that's actually representative of what we see in people living with the disease. And, this is how I spend most of my time. This is what's funded by the CDC, by the CDC ALS registry. And the remarkable thing is that because of the funding of the CDC registry, and by the way, this is Kathleen Wilsbach and Catherine Gallo. They do all the work. Nothing would be possible without them. They're just unbelievable. Dr. Hyman Patterson, who's talking right now about another topic, is started and directs the ALSO Foundation, and is my partner at Temple. And she had 30 autopsies that she had collected over many years that she very graciously donated to this effort as well. And we spend our weeks on Zoom, and this has been for a long time, way before the pandemic, working with researchers. And when we are talking to these researchers, again, just as Dr. Griffin had first suggested, we're not worried about intellectual property or authorship or ownership. In fact, we don't want any of that. We don't ask to be put on papers. We don't ask to be, given credit or have IP. And we're only able to do that because of the support we get from the CDC, right? And so when we get on these Zooms, we can just focus on what can we do to help? How can we help move this research forward as best as possible, as responsibly as possible? Make sure that even at the earliest basic science, they're thinking about biomarkers for eventual clinical trials. Make sure that these precious samples that are donated by people who had ALS and their families are not wasted. But we say yes to everybody, and we do whatever we can, and it's only possible because of the funding that we get from the CDC. And maybe some people who are going to view this video or are on the Zoom right now have had these calls with us because we are meticulous and rigorous about this stuff. And we don't just give these samples out until we're sure that these studies are well designed, and how are they going to be interpreted, and is there a go no go decision for developing a therapy, or is it the right sample set? And we do whatever we can to help make sure that these experiments succeed. And so that's actually how I first became involved with that study that I showed you of those mice and the MG53, because that was a study in these mice, right? And so this is mouse muscle, mouse diaphragm muscle that makes you breathe. This WT, this is normal mouse muscle. This one here, this is that mouse with the SOD mutation, the ALS mouse. And this is a stain for a particular thing related to this pathway. And I think you can clearly see the difference. This is also muscle. These are muscle fibers. These are cut on end. These are looking at them, sort of lengthwise. And we contributed autopsy tissues from people who live with this disease to see if we saw those same changes, right? So this is normal people who don't have ALS. This is sporadic ALS. Again, looks very similar. This is, C9, a type of genetic ALS. And so this is how we use these kinds of resources to validate what we see in the animal models. Last year, this gentleman, Dr. Garudo, reached out to the CDC. Dr. Garudo had been curating this huge collection of blood and spinal fluid and autopsy tissues from people who had died during the epidemic of what was called Lydico-Bodig on the island of Guam over many decades, and were collected by NIH, right? And so, the island of Guam had this epidemic of this disease where some people got what looked like ALS, some people got something called Parkinson's dementia complex. At one point in the 40s and 50s, 10% of and I think 60s too, 10% of the population of the Chamorros, the people living on Guam, were dying of ALS. Another 10% were dying of Parkinson's dementia. They collected all this rigorous data, their family members' data, serial blood samples over time, in some cases blood samples from people who started out normal and then developed disease, their autopsies after they died. Dr. Garudo approached the CDC and said, hey, I'm retiring. I would like this to be used by researchers. Could you use it? CDC reached out to us and said, hey, can you incorporate this into your resources? And we said, of course. And they graciously funded what was necessary for us to do so. And so now we're in the process of analyzing all these samples and to make them broadly available. And having these blood and spinal fluid samples matched to the tissues really is the thing that's going to help us figure out, are changes that we're seeing in blood or in spinal fluid during somebody's life, how do they relate to what's seen in the autopsies? And more importantly, this epidemic was thought to be somehow a cross-section of an environmental exposure, right? Toxins, maybe something they were eating, maybe something that they were including in their diets or exposed to in a genetically susceptible population, possibly. And so these samples are going to help us figure that out. And they're going to help us figure out, how does ALS relate to Parkinson's disease and these other disorders? And so I'm really excited to get these into the hands of researchers. By the way, they came with all these paper records. Nothing's computerized. It's an unbelievable chore. If anybody lives in Philadelphia and wants to come help us, please, we need help. They came with all the raw data of zillions of experiments done on these samples, which we are now annotating to the samples themselves so that we can share that data with researchers. All kinds of studies on all sorts of stuff that use these tissues over years. And all of this is going to be made publicly available. So again, what are the things that I'm most excited about and most my wish list of what I'd like to see over the next year, let's say, or the next few years? And so one of them is the way that personalized therapies are advancing. We are identifying more genes. We are figuring out how to target genetic ALS. And if you have a mutation, perhaps therapies that are going to work. But we also need to be more personalized about the non-genetic types of ALS and making sure that we have mechanism-specific biomarkers to identify who should be in which trials, because that's the only way these trials are going to work. Okay. This is something, that second item that's listed there, is my own personal wish. Again, I don't speak for the CDC, but the CDC registry was not conceived in a time in this data-heavy world that we live in today. And if I had my one wish, and by the way, this idea came from a person living with the disease, an advocate who suggested it to me. I wish that the CDC had additional funding to administer a truly global unique identifier for ALS research. Because right now, the biggest barrier to sharing data and sharing samples is that these things were all, it's hard to do so respecting the making sure that it stays de-identified and all the regulatory concerns related to that, right? And there's different ways that the samples are de-identified and different, codes that are used, and all of our institutions interpret it differently. And it's an unbelievable headache for researchers, for clinicians, and we just want to share it. And people living with this disease want us to share it. And, you know, I think it would be wonderful if the CDC could had, you know, the bandwidth, the funding to administer a truly unique identifier for ALS research. And then, as I said, you know, I would love it if we had the ability through granting mechanisms, through the DoD, through otherwise, to help improve the design of later stage trials. I don't think that we should tolerate trials targeting a specific mechanism developed in a specific lab model without A, knowing which people living with the disease are the right ones to enroll, and B, having the tools to see if the biology is changing in that pathway in the people. And guess what? We are getting better at that, and it's happening. Right now, there are many trials that are doing this. There are trials looking at, you know, different types of inflammation that are measuring markers of inflammation in the spinal fluid as a requirement to get into the trial. There are trials of something called endogenous retroviruses, something called HERV-K, which are using a blood test to look at DNA levels of this, you know, this HERV-K, this activation of these things before enrolling people into the trials. That needs to become the norm. It needs to be something that we're doing. You know, again, we would never have expected the SOD Qalsody trial to work if we enrolled everyone with ALS, and we need to be doing the same thing for these sporadic trials. And then the other thing at the end there, and I showed End the Legacy before on my first slide. So, you know, we have this massively growing community of people with genes at risk for these diseases and who haven't yet developed them, right? So people who have, you know, SOD, C9, all these mutations, but don't have the diseases yet. And we need to learn how to follow them, what we should be measuring, how to do clinical trials. If I have a gene therapy, I'd love to give it to somebody before they have ALS, right? How do we do that? How do we know if it's working? What do we measure? And the Legacy is just, check them out. They're doing an unbelievable job of kind of leading the way and also providing educational materials and, you know, for example, considerations on whether you, on what to know, risks and benefits of getting gene tested if you're a family member of somebody with ALS. Check them out. But, you know, we need to figure out how to do this research. We need to figure out how to monitor these people. I personally think that the digital markers are going to be a panacea for this, because we can, you know, measure somebody's strength with a digital, like a Bluetooth, you know, squishy ball that measures their grip, and they can have it in their home and they can squeeze it once a week and see whether or not that's changing. And we couldn't bring all these people into our clinic. So I think the future looks bright. Last thing I want to mention, we need to work together to be responsible consumers of information. Every day somebody sends me some article like this, right? Breakthrough can halt the progression. And I'm like, oh, what's this? And I go, look. And by the way, these are amazing scientists, and it's amazing science, and it's really, really exciting. But you know what it's not is something that's going to necessarily halt the progression of ALS, or at least we don't know yet, because the data in that trial, look at this. This is like that picture I showed you of the mouse originally in that MG53 study. This is, you know, they're not living any longer. It actually says right here, where is it? Somewhere in this, no survival improvement was observed, right? So this is a really remarkable study showing that they can do a particular kind of manipulation and that it's safe and that it appears to be affecting some mechanisms in a way that could someday lead to therapies. But the way that it was interpreted by the public press was you know, not responsible. And nowadays we have something called preprints. So you guys get to see articles that are before they're even peer reviewed and published, right? Before anybody's seen them. And, you know, we have to learn to be responsible consumers. If anyone here's done, you know, the Clinical Research Learning Institutes through NEOs, which are amazing. Part of what we do is we say, you know, we teach how to read a paper, right? How to read a grant application so you can be good grant reviewers for the DoD. And then the community can, you know, spread that wisdom and teach that to others. And, you know, there already are people like, for example, Nadia Sethi is unbelievable. You know, there's a ALS Clinical Trials Facebook group that she moderates that's helping with a lot of this. But this is the way that we need to work together to spread the knowledge of how to consume this information. And the analogy that I use is, once upon a time, everybody got around in horse-drawn buggies, and then cars were invented, right? And much like preprints spread data quicker, cars were a lot faster. They got you from point A to B super fast. But those first cars blew up, and they crashed, and they killed people. And we had to learn how to drive. And so, you know, nowadays we all get driver's license and we have to go through driver's ed. And I have a 20-year-old son, and I'm really glad that he had driver's ed and I didn't have to teach him. Somebody who's trained to teach him taught him how to drive a car. And so what we need is the equivalent of driver's ed for this data heavy world we live in, to teach us how to consume information responsibly. Last slide I want to show. So I started off the last time I spoke talking about one of my mentors, Dr. Griffin, who had passed away and was, somebody who was really important to teaching me how to be a neurologist and be a clinician. Very recently, another one of my mentors, this is Fred Sachs. He was my PhD mentor. He passed away a couple months ago, and because of this interconnected world we live in, a lot of us who haven't spoken to each other in a really, really long time started sharing stories on Facebook with each other and got back in contact with each other and talking about how he impacted us. And I just wanted to share it because when I think about what I'm personally going to focus on in this next year, it's time to figure out selective vulnerability. It's time to figure out how all these diverse mechanisms and genes, how all these different things can legitimately cause ALS, and yet you get this very specific disease. And I think we can do it. I think we have the tools and the technology, and I would just tell you, stay tuned. I've dedicated my life to this disease, and it's because of you and people living with the disease that I've done so, and I don't intend to fail. So thank you guys very much.
Thank you, Dr. Ostrow, for that great presentation. It is wonderful to hear that just in the last year, as when you spoke at the Engage Symposium, that now there's so much more to talk about and we can continue to have this conversation. And I think that image of your father-in-law in his footy pajamas will stick with me for a long time to come. So thanks so much. We do have just a few questions, if you don't mind sticking around with us.
Let me add one thing too, which is that I'm happy to talk to anyone anytime. You guys email me, reach out on social media. I work for you, right? Anyone on this call, anyone who watches this video, I mean it. If there's ever anything I can do, this is the reason for this. So please don't be shy.
And we do appreciate your dedication to the neuromuscular community and to your support with MDA. So if we can just take a few questions, we did have a few folks who put in questions before the webinar. One of which was, can you talk about stem cell research in ALS? Just touch on that a little bit more.
Sure. So there's two different ways that stem cells are used in ALS. And by the way, the word stem cell means slightly different things to different people, right? And so the basic idea is that there's some cell that we've taken, whether it's from skin cells, blood cells, fat cells, and been able to turn it into a cell that is able to then become other kinds of cells, and we can make them into brain cells, right? Into neurons, into other kinds of cells in the brain, whatever they might be. And so you might hear a lot about what are called iPSCs, induced pluripotent stem cells, which is what that is, and then we can make those into nerve cells. And in the lab, we can do that, and we can take cells from people who have ALS or different flavors of ALS, genetic ALS, fast ALS, slow ALS, and we can compare those to people who don't have ALS, and we can look at what's different. And so they let you sort of do a clinical trial in a dish. And so they're one of the patient-derived tools that we think is going to help us validate findings in, like in a mouse, in something that's more relevant. Now, as far as using them for therapies go, I truly believe that in my career, at some point, I will use stem cells that are somehow engineered to create, for example, certain growth factors in treating ALS. But here's the problem. And so far, in the examples that we have of this, they really appear to work for some people, but clearly it's some people, right? And again, it gets back to what I was stressing earlier. We need to figure out who those people are. We, just like any other mechanistic therapy in ALS, we need to have a way to tell who are the people that should be enrolled in a trial of a particular stem cell therapy. And those are the people that should be enrolled. And it's not just a matter of that's the way we're going to get successful trials. It's also because, things that are infusions or require this sort of like taking cells out and doing something and putting them back, I mean, it's a lot of work, and our clinics are stretched thin, right? We don't have the bandwidth, and, MDA certainly supports, clinics around the country and helps us do what we do. But if we can't figure out who we're supposed to give which therapies to, it's going to impact in a detrimental way our ability to take care for other people, right? Because it's going to take a lot of resources and a lot of time. And so I really want every therapy to work. And I know that some of these stem cell therapies appear to be very exciting in some people. We have to have predictive biomarkers that can tell us who those people are.
Yes, so much variation. Thank you for that. So we do have just one or two more questions for you. One question that we had from the audience was, now that Relivrio has been pulled from American markets, what new treatments or medications do you have hope for in the near future?
Right. So, Relivrio, I still believe it probably works for some people, and I wish I knew who they were. And I think that maybe the story's not completely over. I know the drug's been pulled from the market, but I know that they're still looking at things like survival data that hasn't been collected yet and some other things. And, like all of these therapies, I think if we can figure out which people should have gotten that drug, maybe there's some hope for it. Now, the therapies again that I'm most excited for is all of them because I want everything to work. And as I told you, I feel that it would be disingenuous of me to tell you that I think one's better than another. There's so much going on. And any ALS expert who, like, I can tell you about the biology. I can explain to you how each of them works. I can tell you how the trial's designed. I can tell you why we think it's a good idea. But I don't know what's going to work. And if I did, or if my colleagues did, we would have been better at translating these diseases. And so what I would say is we depend on people living with the disease for clinical trials. We have improved the way we do clinical trials with your help doing, clinical trial design that is responsive to people living with ALS and including them and working towards better care equity and sharing placebos and the Healy trial and all these unbelievable changes that are happening to the way we do trials. And so the ones that I'm excited for are the ones that have biomarkers that are going to tell us, are we enrolling the right people? And so some of those examples, for example, there's the baricitinib trial right now that's going on at MGH. The rare gene trials that are happening at Columbia, the thing that's called Silence ALS, that those are only for ultra rare mutations. I mentioned the HERV-K trials, which are using a marker in the blood, but the list is long. And it's anybody that's really rigorously trying to figure out which people to give the drug to and can they detect the biological change, because the ones that I don't want to hear about anymore, I'm tired of trials that end and we don't know why they failed. We don't know if it was a good idea. We don't learn from them. And so beyond that, I want them all to work.
Yes, even in those failures, we can learn so much. So it's important to take that into consideration. Just one more question for you before we let you go. How do environmental factors potentially contribute to ALS? And how is this aspect being explored in research?
Right. So, and by the way, one other comment about the trials, that website that I shared, that Google Doc at the beginning, that's my personal go-to for updated information on all the trials. And so check it out. And all the publications and all the research, it's really a remarkable resource. We know that somehow that ALS involves, some kind of environmental interactions in people who may be genetically susceptible, right? And certainly the epidemic on Guam is the most famous example of that, where it was just clearly there was something about what they were eating or exposed to, and there's a bunch of theories that may or may not be right. But right now we have these resources to finally answer that question, and the CDC in particular is kind of leading the way. I think that we need to get better about doing this remote data collection and involving everybody in these studies. And so, as an example, Dr. Hyman Patterson, who I mentioned, is giving a talk right now too. She's been looking at diet-related factors and, through a survey, trying to figure out, are there particular diets, particular types of nutrition that might be impactful? And that diet-related survey is now going to be incorporated into the Everything ALS app, right? And so, a digital app that can be used by people around the country and hopefully will help us gather this data. So by sharing this kind of data, by making it broadly available, by putting it in ways that people, whoever you are, wherever you live, can contribute, is how we're going to answer that question. And really importantly, for the people with these gene mutations who don't yet have ALS, we call, at people who are at risk because they have a mutation, like those are the people. I mean, if I had an ALS gene mutation, I would want to know, like, does it matter what I eat? Does it matter where I live? Should I move to a different climate? What kind of exercise should I do? Right. And these are the efforts that are going to help us figure that out. And again, these efforts are being driven by the community, not by a particular person or a scientist. A lot of it is CDC, but also organizations like End the Legacy and, all these groups, ALS TDI, Everything ALS, I Am ALS, that are doing all this digital work too.
Yes, great. Yeah, the stronger our ALS community, the more answers and support we can get in the long term. So thank you so much for that. And I think we're going to wrap up questions now, but I would like to thank you again, Dr. Ostrow. We sincerely appreciate your time, expertise, and everything you do for the ALS community and the neuromuscular disease community as a whole.
Thank you so much. And again, if anyone wants to talk ever, don't be a stranger.
Thank you so much. We are now pleased to welcome MDA's Vice President of Public Policy and Advocacy, Paul Melmeyer. In this role, Paul leads MDA's policy and advocacy initiatives pertaining to public health, therapeutic development, access to care, and disabilities. Prior to joining MDA, Paul spent over 6 years with the National Organization for Rare Disease. At NORD, Paul led the federal policy operations in developing and advocating for the enactment and implementation of pro-rare disease patient policy. Paul also holds a Master of Public Policy from George Washington University. Paul is constantly seeking opportunities to make a positive difference through compassionate and effective policy change. Thank you so much for joining us today, Paul, and I'm going to hand it over to you.
All right, well, thank you very much, Tess, and good afternoon or evening, everybody. I am Paul Melmeyer, Executive Vice President of Public Policy and Advocacy. Before I get started, I'm just going to share my screen so you all can see just a brief presentation I put together on essentially what's going on in public policy and advocacy at the intersection with ALS. So let me get into presentation mode here. Very good. So as Tess mentioned, I lead the Public Policy and Advocacy Department within the Muscular Dystrophy Association. And really everything we do within MDA's Public Policy and Advocacy Department falls within 3 general areas of public policy making. The first is where we try to accelerate therapeutic development for those with neuromuscular conditions, trying to get more and better treatments to the community more quickly than they're arriving today. The second is to increase access to healthcare for those with neuromuscular diseases, and that may be access to specialists or multidisciplinary care clinics, access to approved FDA therapies, medical devices, power wheelchairs or mobility devices, access to telehealth or quality health insurance, really just anything that our community utilizes to manage their health. We want to be knocking down barriers to accessing those services. And then similarly, we're also looking to break down societal barriers that still remain in front of those with disabilities, and that includes access to accessible transportation or to an education or to employment or a career, or just really anything that a member of our community might want to pursue, but due to oftentimes society's backwards orientation or structuring, thus disadvantaging those with disabilities, there are a number of reasons for why folks in our community might not be able to do so. So we within the Public Policy and Advocacy Department strive to pass public policies, enact public policies that really kind of fit within these 3 areas. And it's no different when we look specifically at ALS. We, of course, try to advance research and drug development within ALS, try to broaden access to care and services for those with ALS, and then break down societal barriers in front of those with ALS as well. So what I'm going to do for the next 10 minutes or so, let's say, is just go through what we're doing in all of these areas, what you should know is happening in Washington, D.C., in efforts across these 3 broad pillars of our advocacy, and also a handful of examples of how you can join us. At the conclusion of the presentation, when I stop sharing my screen, I'll put some opportunities in the chat box for you to join us on efforts that actually span all 3 of these areas. To call your congressperson or to write to the administration on things that we believe could be very impactful for those in the ALS community. So I want to start with our efforts to advance through public policy making, research, and drug development within ALS. And I want to start kind of at the research level of things, and at the National Institutes of Health, because there's a lot that the NIH is doing that not only they have been doing that could be very impactful within discovering some of the things that are still either undiscovered or need better understanding in ALS, and that includes basic and translational research, some of which I heard Dr. Ostrow talking about here, but then also other areas in which NIH is active in trying to better understand ALS and actually bring new potential therapies to the biopharmaceutical industry for them to develop. Some of the more specific things NIH continues to lead are the Expanded Access Grants Program that was commissioned as part of the Act for ALS, in which $75 million per year is appropriated to this program to offer expanded access to certain therapies that are within phase 2 or 3 clinical trials that then otherwise individuals with ALS may not be able to access unless they're actually participating within that clinical trial. The Act for ALS was passed just a handful of years ago, and there are additional provisions within that bill I'll get to here momentarily. But this provision specifically for the Expanded Access Grants Program continues to move forward strongly, with $75 million per year being appropriated for the last several years, including within the most recent appropriations bill that Congress passed just a little over a month ago at this point. The NIH is also pursuing, along with the Friends of NIH and other partners, something called an AMP program, in which they're really trying to kind of revolutionize the approach to doing research within ALS. And then also within the last couple of years, they actually went through a process to update the strategic plan for research in ALS, and now they're in the process of implementing said strategic plan. Appropriations to the NIH continue to go fairly strongly, while there are some who wish to reduce appropriations to the NIH. Thankfully, that has not happened, and in reality, NIH continues to fund millions of dollars of research into ALS every single year. So moving on from the NIH briefly to the Food and Drug Administration, the FDA is also moving forward with several initiatives, some of which come again from the Act for ALS, that are we believe to be impactful in bringing new therapies potentially to market for those with ALS. The first two I want to mention are the Rare Neurodegenerative Disease Action Plan and the Rare Neurodegenerative Disease Grants Program, two programs that were commissioned as part of Act for ALS. The action plan is a regulatory action plan in which FDA, back in the summer of 2022, issued an action plan that laid out the different regulatory priorities over the ensuing 5 years that the agency would undertake to try to accelerate the development and subsequent approval of new therapies for ALS. And actually, we just checked in with the FDA on this action plan last week, and sure enough, they are making strides in accomplishing several of the things within the action plan. And I'll get back to one of those here momentarily in partnership with CPATH. But also at the same time, they're also now, I believe, in the third year of the Rare Neurodegenerative Disease Grants Program, in which they give $5 million of grants to researchers researching rare neurodegenerative diseases to try through that research to accelerate the development of new therapies for rare neurodegenerative diseases, including ALS. And some of the grants in the past that have been funded by this program include efforts to actually allow for the ALS FRS endpoints to be used in a decentralized clinical trial, i.e. be used at home. So those with ALS don't have to travel to centralized clinics to participate within clinical trials that use the ALS FRS as the endpoint. And then there are also studies that have to do with other aspects of usage of certain advanced devices within ALS, as well as studies outside of ALS in myotonic dystrophy, in some ataxias, in some other neurodegenerative diseases, all funded by this grants program. And again, this was appropriated at least $5 million, and we're still trying to get clarity if they actually got a few $1 million more this year. I think they might have actually got upwards of $7.5 million for this grants program for this fiscal year from Congress. Now, also happening at the same time is a public-private partnership commissioned by the Act for ALS that is being led by CPATH. CPATH is the short name for the Critical Path Institute, and this is an entity that seeks ways to accelerate therapeutic development across a variety of diseases. But they were commissioned by the FDA and NIH to run the public-private partnership that was commissioned by the Act for ALS. And they've already made a good amount of progress. And one of the things they wanted to come back to that I mentioned through the action plan is that CPATH, along with the FDA and the NIH, are announcing efforts to accelerate the development of alternative endpoints for clinical trials. And these endpoints could be incorporated in clinical trials going forward so that there may be alternatives to the ALS FRS within clinical trials in the not so distant future. Most clinical trials right now, especially pivotal clinical trials within ALS, use ALS FRS. And for those who aren't familiar with that acronym, that's ALS Functional Rating Score. It's kind of a composite of different functions and measuring of functions in those with ALS to try to come up with a overall meaningful evaluation of whether an individual with ALS is progressing as quickly as they would have otherwise if they're happening to be in a clinical trial. But there are challenges with the ALS FRS. It's not perfect. And so there's a new effort to try to develop new endpoints, new clinical outcome assessments within ALS so that all of the clinical trial eggs, if you will, aren't in the ALS FRS basket. And so those are just a handful of examples on the FDA side of things of ways in which they're trying to accelerate therapeutic development in ALS. And Congress is also, to some extent, doing their part. It's actually a fairly quiet year on research and therapeutic development, public policy making in Congress. Nonetheless, there are a couple of bills that they're working on, including a bill that's called the Benefit Act that seeks to incorporate patient viewpoints in FDA's decision making more so than is occurring today, as well as a bill called the Promising Pathway Act that would actually create a whole brand new conditional approval pathway at the FDA, which is still very much in draft form. It's still not quite ready for prime time, if you will, but is being worked on and is being supported by a number of those within the ALS community. Moving forward to the second area of public policy on broadening access to care and services. Here again, there are a number of active efforts to try to deliver more and better care to those with ALS. The first that I want to highlight is an effort to pass the ALS Better Care Act, and this is being co-led by ourselves working with the ALS Association and IMALS. Now, the ALS Better Care Act would actually create a bundled payment system for ALS multidisciplinary care that would then strengthen reimbursement and access. And the reason why this is important is that at multidisciplinary care clinics, and I imagine many of you who are watching who have ALS or as a family member of someone with ALS, you may be familiar with the model of multidisciplinary care for those with ALS at this point. But the model is essentially an individual with ALS will head to the clinic and spend much of the day there seeing many of the specialists who happen to be at that clinic. So not just a neurologist, but maybe the dietitian, the genetic counselor, and the physical therapist, the occupational therapist, the registered nurse, the list of specialties, the pulmonologist, the rheumatologist, I don't know, just the list of specialties involved in caring for those with ALS is a long one, frankly, and many are present within ALS multidisciplinary care clinics. However, much of that care actually goes uncovered or unreimbursed. Oftentimes, it's only the neurologist and the nurse who actually has their care covered by the Medicare program or by private insurance, depending on what insurance the individual with ALS happens to have. And consequently, the clinic has to eat all of the costs of providing that genetic counselor or the dietitian or the physical therapist or speech therapist to the individual with ALS. And that's not really a sustainable system. That makes it difficult for the clinics to actually provide these services, and sometimes some clinics actually have to go without providing these services. And if those services are indeed provided, sometimes there's just a shortage of the specialists to offer to those with ALS, making it more difficult to actually access those individuals. So this bill would solve that, at least within the Medicare program, by creating a bundled payment system in which all of these services would be covered and reimbursed for by Medicare. And consequently, any challenge that the clinics may have in providing these services to the community through the Medicare program should be obviated at that point, which would be very good news. And so we're working with these organizations to try to get this bill passed. And at the conclusion of the presentation, I will put within the chat how you can join us in trying to encourage Congress to pass this bill. We're looking for co-sponsors of this bill. And so that means that your representative is probably not on the legislation as of yet. They probably haven't signed their name as a supporter. And so we'll ask you to ask them to support the ALS Better Care Act. But this isn't the only bill to mention that's within this general area of policymaking that Congress is tackling. Additional efforts include efforts to continue telehealth flexibilities and the accessibility of telehealth. That technically will end at the end of this year. These pandemic era flexibilities in using telehealth that were passed just a handful of years ago That will end if Congress doesn't act by the end of this year. We're expecting Congress to act, but it's not guaranteed. And so this is something that we're working with Congress to actually make sure happens by the end of the year so that everybody within our community can continue to access the telehealth that they have found valuable to access. There's efforts to expand access to home and community-based services, the critical services to allow our community to stay within their home, to remain independent within their home, but still access home healthcare aides or other attendants who help with activities of daily living. But we frequently hear from those within the ALS community, those within many of the communities that MDA serves, that home and community-based services are very difficult to come by. They can be expensive or not covered if they're accessible even in the first place, that there are shortages of individuals who provide home and community-based services, and consequently something needs to be done about this. And there are several bills that we're working up on Capitol Hill on to try to address this issue. Finally, the last thing to mention is access to genetic counseling services. I already mentioned that genetic counselors oftentimes are not covered by the Medicare program, and consequently the ALS Better Care Act would in some ways solve that by offering a bundled payment for all the services that are offered by multidisciplinary care clinics. But we're trying to address for genetic counseling services the issue more broadly, because it's not just those with ALS who can benefit from having a genetic counselor offer their services. It's frankly many, if not all, individuals within our community who can benefit from genetic counseling services being more accessible. So we're also trying to pass legislation that will address that very issue. Moving over to the third and final area, and this is our efforts to break down the societal barriers that remain in front of those with ALS in living a full life. And that could be in accessibility issues, that could be accessing in education, that could be accessing employment, that could be a variety of things that society is still constructed in a backwards manner to disadvantage those with disabilities. And that, of course, includes those with ALS. The thing that we've been focusing on first and foremost, certainly this year, and actually it's happening almost as we speak in the Senate, is efforts to make air travel more accessible. We hear frequently from the community that air travel is incredibly inaccessible and it's unsafe and frankly quite undignified for a wheelchair user to go through the commercial air travel system and having to endure seat transfers, thus potentially injuring folks in our community and having to risk wheelchairs or other mobility devices being damaged as they're being stowed in the cargo hold of airplanes. And we hear far too many stories, 10,000 wheelchairs every year damaged by the airline industry. So the good news, though, is that we're very close to addressing a lot of this through public policy making. And the thing that's happening, as I said, almost as we speak, is a bill called the Federal Aviation Administration Reauthorization, the FAA reauthorization. The FAA is an agency that needs to, they and their authorities need to be reauthorized every 5 years. And Congress uses that opportunity to really take a close look at pretty much all aspects of commercial air travel and reform anything within that system that needs to be reformed. And clearly the inaccessibility of commercial air travel is one of the things that needs to be reformed the most substantially. So the good news is that we're at the point in which the House has already passed their version of this bill that included a variety of accessibility provisions within their legislation. And the Senate is debating their bill literally today. They were debating it yesterday, they've been debating it this week, and they're so close to getting across the finish line. They might pass it tomorrow, in fact, for that matter. And if they do, then that bill, which has actually been merged with the House bill, would still need to be passed by the House again because it's different than the version they passed. Actually, it was all the way last summer, almost a year ago at this point. And then in addition to that, the president would have to sign the legislation as well. But we're expecting all of that to happen in the coming days or perhaps in the next week or so. And so consequently, we are this close to having one of the most substantial improvements to accessible air travel in nearly 40 years passed by Congress. I actually haven't told you all that much of what's in the legislation as of yet. I'll just mention a few things right now. For one, and perhaps most importantly, the pathway to actually having wheelchair spots on airplanes is delineated within this legislation. So there are still some technical and economic feasibility studies that need to be concluded to check all the boxes that the Federal Aviation Administration needs to see to ensure that wheelchair users and those around wheelchair users can fly safely within their wheelchair. So that research would require to be completed within the next handful of years. And then from there, the Department of Transportation would have to issue the plan for how wheelchair spots on airplanes will come to fruition within the coming years from there. Also within the legislation are very robust training requirements for baggage handlers so that they will require to go through hands-on training to ensure that they know how to fold wheelchairs or move wheelchairs or just stow wheelchairs properly within the cargo hold of an airplane. And then there's also hands-on training required of airline or airplane personnel who conduct seat transfers, so that individuals who are untrained and unknowledgeable on actually how to interact with an individual with a disability, especially someone with a medically complex condition such as ALS, and who's actually going to be transferred from one seat to the other, that clearly needs a lot of training, and yet individuals right now don't have to be trained on this. That would change with this legislation, thankfully. And then also in the legislation, there'd be new evacuation standards for those with disabilities. There'd be new accessibility standards for airports and also airplanes themselves. There would be requirements for onboard wheelchairs to be on airplanes. And from there, additional provisions, frankly. I can't get into all of it in the short amount of time I have, but it's pretty substantial, and we're very excited about it. And again, I will, at the conclusion of my time here, I'll put into the chat how you can encourage your senators and representatives to pass this bill here in the coming days. Now, while this is happening, the administration also is working on a proposed rule that would also make air travel more safe and dignified for those with disabilities. And while there's some overlap with the effort going on in Congress, there's also provisions within this proposed rule that would actually require airlines to return and replace damaged wheelchairs under a certain time frame, that would require loaner wheelchairs to be more specialized for those with disabilities who had their wheelchair damaged and is being repaired or replaced, and consequently are using a loaner wheelchair at that given time. There's provisions on defining what safe and dignified air travel actually means. And there's a number of other provisions within the proposed rule that we're very excited to see within this proposal. Now, the deadline the administration has set for this proposal has actually been extended. Initially, it was May 13th, just a few days away. They've extended it to June 12th. And that's good news, because that means we all have just a little while longer to support this proposed rule and really encourage the Department of Transportation to finalize it. So here again, in a moment, I will put into the comments how you can tell the Department of Transportation why safe and dignified air travel is important to you. And we do encourage you to write in and share your personal stories. If you have gone through the unfortunate experiences of experiencing inaccessible or even dangerous air travel for that matter, please share that personal story with the department. They want to hear those stories. It's those personal stories that really make a difference. And actually that picture in the center of your screen there from February 29th, when I and a number of other advocates joined the Department of Transportation. You see Secretary Pete Buttigieg there, as well as Senator Tammy Duckworth. It was at that very announcement of this proposed rule that Secretary Buttigieg was highly encouraging of the community to tell personal stories to help inform the department as they're finalizing this rule. I'll just close by saying these aren't the only efforts ongoing within are trying to break down the barriers in front of societal barriers in front of those with ALS. We're trying to reform benefit programs such as supplemental security income and Social Security disability insurance. We're trying to increase accessible housing. We know there are shortages of accessible housing out there. There's a bill called the Vital Act that would try to address this, and we're supportive of that bill. And then also broaden educational opportunities, employment opportunities. There's a number of other efforts happening within this area. But by now, I'm sure I probably ran out of time. So I think I'm going to stop there. And I'll welcome any questions anyone has. And similar to Dr. Ostrow, please reach out to me anytime as well. There's my email, pmelmeyer@mdausa.org. We're here to serve you. Our public policy and advocacy team is here to serve you and try to address what you might be experiencing through public policy change. And so reach out anytime and please join us. And again, as I said, when I stop talking here and stop sharing my screen, I'll put those opportunities for emailing your congressperson as well as contacting the administration on their proposal for accessible air travel in the chat. And with that, I welcome any questions.
Thank you so much, Paul, and thank you for the important work that you do. And it's really not possible without the support of the community as a whole. So we do encourage you to get involved if you can. I do also have some slides to share and ways you can get involved. But again, if you have any questions for Paul, you can reach out to him directly, or you can also reach us at advocacy@mdausa.org. And Paul, feel free to stay on, and if you see any of the questions in the chat that you want to go ahead and answer, you can go ahead and feel free to do that.
Yeah, thank you, Tess. And just to highlight these two opportunities in front of you here, and Tess and Marissa, thanks for putting this together. If you scan the top QR code there, that is just to join us overall within our advocacy efforts, and you'll get all of the updates that I've talked about here in your inbox, so that you can take action with us as soon as those opportunities are ready. And then in addition to that, we actually have, in acknowledgement of ALS Awareness Month, an Advocacy Institute next Wednesday evening at 7 p.m. Eastern Time. We're going to be especially focusing on ALS Better Care Act, but also the implementation of the Act for ALS, joined by someone likely from the NIH talking about their work there. So that's the bottom QR code there, the one that looks a little bit messier, if you will. Feel free to scan that and join us for our Advocacy Institute next Wednesday evening, 1 week from today.
Yes, thank you. We do invite you to join in our advocacy efforts, or just to continue to learn more about policies impacting the ALS community. So check out those QR codes, and hopefully you can join us this Wednesday, May 15th at 7. All right. Do you see any questions in the chat for you there, Paul?
I don't see any questions in the chat, so Tess, I think we might have a quiet audience for the evening, and that's quite okay. I can hand it back to you.
Absolutely. Well, your presentation was very thorough, and you gave us a lot of great information. So thank you for being here tonight. We appreciate it. Right, now it's my pleasure to introduce our next speaker, Sarah Dramstad, RD, CD, CNSC. Sarah is a clinical dietitian at the University of Vermont Medical Center in Burlington, Vermont. She has previously worked as a clinical dietitian at the Mayo Clinic in Rochester, Minnesota. Nutrition support has always been an area of interest to Sarah, and to further enhance her nutrition knowledge and improve the quality of care for her patients, she has obtained the Certified Nutrition Support Clinician Certification. Thank you so much for being here today, Sarah. I'll now turn it over to you.
Thank you, Tess, and I'm going to share my screen. All right, so let me know too if I need to speak up a little bit louder for my microphone, but I'll go ahead and get started here. So I just wanted to thank everyone tonight for joining in. Like Tess said, I am Sarah Dramstad, and I'll be talking about nutrition considerations in ALS with the collaboration of the MDA. So again, briefly about me, I am a registered dietitian. I'm certified in nutrition support, currently working at the University of Vermont Medical Center in Burlington, Vermont, and then I previously worked at the Mayo Clinic in Rochester, Minnesota. So tonight's presentation objectives for you all is to further understand what role does a dietitian have for those with ALS, to understand the impact of diet and nutrition on symptom management and daily living for those with ALS and their caregivers, to understand important nutrition considerations for those with ALS, and to provide resources and information to help those with ALS and their caregivers make informed decisions about their care. So I'll start out with, what can a dietitian do to help you? So when you go to a clinic, we start by asking a series of questions, and this helps get to know you, as well as have us be able to have a comprehensive nutrition assessment. So we may be asking about your diet, what you typically eat, your weight trends, food allergies, preferences, and further more that I'll get into the next slide. But this is really for us to understand where your nutrition is at, where your diet is at, and help identify any gaps and where we can help improve your nutritional status. And specifically for those with ALS, I'll talk a little bit more in detail about what that really means. We do work really closely with a speech language pathologist or an SLP to modify that diet when chewing and swallowing become more difficult. Because as you know, as ALS progresses, it starts to affect those muscles involved with chewing and swallowing. And so we work together really closely to ensure that you're able to have those safe food textures and comfortable textures for you to swallow, as well as making sure you're meeting your nutritional needs. Now, dietitians will also talk to you about and explore the option of using oral nutrition supplements. We'll also talk about exploring the option of using a feeding tube. But really, I want to highlight making sure that we help promote the enjoyment of food and eating and work with you individually to help meet any of your nutrition goals of care that you have. So what exactly does a dietitian assess? Like I said, it's a comprehensive assessment, so we will be asking a lot of questions. But again, we start out with just gauging about your diet history. So we may ask for a 24-hour recall or just asking you to describe what a typical day of eating looks like. I know that we all eat something differently every day, but it's really just to help identify the types of foods that you eat, your appetite, if you're more of a meal person or if you'd just like to snack throughout the day. We take a look at any of your eating patterns. We'll also take a look at any past diet modifications you've had regarding solid food texture or liquid consistencies. We'll take into account any food allergies, food intolerances, food preferences, cultural practices, or any other dietary restrictions that you may have. We'll take a look at your weight history and your weight trends. We'll assess your hydration status and your bowel movement. And we'll also take a look at your vitamin and mineral supplementation, as well as any other health diseases or comorbidities that you may have. So this may include diabetes, cardiac health, digestive health, or anything else that you think is important for us to know. And then we help in assess your daily calorie and protein and fluid goals. And this is all going to depend on what we find and what we assess based on the questions that we ask. There are different types of equations and methods that we use to help calculate your needs, but starting out with this assessment helps to gauge where you're at. Now, how do we improve your nutritional status with ALS specifically? To start with, meeting your daily calorie needs. People with ALS have a higher need for calories every single day. And this is because of the disease progression. It's going to be more effortful. It's going to take more energy to do things like walking, breathing, eating, and then even non-functional muscle activities such as knee cramps, twitches, if you're experiencing pseudobulbar effect, and then any protein breakdown, especially from muscles that are just not being used. So for those with ALS, it's really important that you're meeting your calorie needs. And that's going to tie into helping to make sure you're maintaining your weight. Weight maintenance is going to be key. A lot of studies show that those with ALS who are able to maintain their weight or even show some weight gain tend to live longer. So making sure you're eating your calorie needs can help maintain that weight. Although we do anticipate seeing some muscle loss in your arms and your legs, if you're able to maintain your weight or even have mild gain, we do anticipate also that you may notice that your body's changing a little bit and you may see some more body mass around your middle or your abdomen. But like I said, for those who are able to maintain their weight or have weight gain, you are at better chance of having a longer lifespan. Monitoring your fluid status to stay hydrated is also going to be really important. Not only does it help mitigate any side effects from just being dehydrated, but again, some studies suggest that staying hydrated and meeting your fluid needs each day can help you live longer. And of course, just making sure to continue to incorporate various food groups and adequate amounts into your diet will just help overall making sure you're having a balanced diet to meet all of your nutrition needs, not just calories, but vitamins and minerals and all the other micronutrients. Now, what happens when you don't meet your nutritional needs? So For those that do struggle to meet their calorie needs, which is often because, again, that calorie need is going to be a lot higher, we will see that weight loss. And when we lose weight, we lose muscle. And when we lose muscle, we're going to see functional loss. And that functional loss is going to further impact your mobility, your respiratory function, and your immune system. So it may be harder to fight off a cold or kind of bounce back and recover. And so we do see an increased risk for malnutrition when it's more harder to meet those nutritional needs. And so how are we going to prevent or treat malnutrition? I'm going to sound like a broken record, but it's going to be eating more calories. Again, eating more calories is going to help your body just maintain your weight. And you may be thinking, what about protein? I mentioned you're having some protein breakdown from not using your muscles. Protein is still important in the diet, and we still want to make sure you're getting enough in your diet, but we don't see as bigger of a gap or bigger of a need to consume more protein as much as we do with calories. So really prioritizing calories is going to be key. And how are we going to do that? So there are many different ways we can add more calories to your diet. A great example is going to be fats. Fats are a great source of high calories. So think of anything like butter, margarine, any oils, nuts or nut butters that you may be able to tolerate, mayonnaise, salad dressings, avocados, fatty fish such as salmon, olives, or even like canned tuna in oil. Anything that's going to contain that fat is going to give you a good source of calories without adding a whole lot of volume to your stomach or making you feel really full. Another example of adding calories to your diet is going to be anything that's concentrated, such as sweet things. So honey, jams, jellies, maple syrup, chocolate syrup, dried fruit, if you can tolerate that texture, but if not, having canned fruit in heavy syrup versus a light syrup or even water is going to give you more calories. Sweetened condensed milk, any creamy soups. Again, it kind of ties back in with the fat. These are all good sources of calories to add anywhere into your meals or snacks that you can, or even beverages. And speaking of beverages, hydration, again, is really important. And if you're able to pick a fluid with some calories, that's great. Think about whole milk, flavored milk, eggnog, fruit juice, lemonade, any sweetened iced tea or sodas, milkshakes, smoothies, and oral nutrition supplements are all going to give you more calories into your diet. And I'll go over some nutrition supplements a little bit later. Other strategies can be adding condiments. So think about any household staples that you might have around. So heavy or whipping cream, sour cream, cream cheese, yogurts, gravy, or even like mixing gravy with some extra fat, or even mix it with yogurt for more calories. Any of the sweet sauces or marinades, such as barbecue or teriyaki sauce. Again, any high-fat salad dressings or mayonnaise-based breads, such as aioli, tartar sauce, pesto, hummus, tahini, guacamole, or any cheese-based dips. These are all really great condiments to add to your diet. If you're someone who likes other types of condiments, such as ketchup, mustard, vinegar, balsamic vinegar, like lemon or lime juice, salsa, hot sauce, soy sauce, those are all perfectly fine to have. They don't contain as many calories, but if it's something you enjoy or something that you like to flavor your food with, by all means, continue to incorporate that as well. Whole fat dairy is another great option for adding more calories. So anything milk-based, yogurt-based, ice cream, cottage cheese, or other types of cheeses that you can add to mixed dishes or to any homemade meals. Again, that fat content is going to give you more calories without making you feel as full to eat as much volume. And of course, treat yourself to any extra snacks and desserts. You're going to make your dietitian really happy by telling them that you're adding an extra snack into your day or having a dessert every day. Because again, it's just another opportunity to add calories to your diet. And of course, enjoy going out to eat. So if you like to go to a restaurant or you like to get takeout, by all means, it's a great opportunity. And oftentimes, restaurant food has more calories, which is awesome. And the best part is you don't have to cook. Other considerations for adding more calories may include smaller, more frequent eating, especially as ALS progresses and that chewing and swallowing just become more difficult, or it just feels more effortful. It's just taking you a lot more time to finish a meal. You might have some snacks or drinks set up nearby, and that just can help prompt you to eat throughout the day in little bits, take a break, but still help you meet your nutrition needs. Having frozen meals readily available is also another great option. So this can be commercially made or even homemade. And it's going to be nice because if you're really tired or you're just really busy, life gets busy, you just don't have time to cook or prepare something, you can quickly pop something into the microwave or oven and just reheat it. Because having a meal or having another opportunity to eat is going to be better than just skipping out. If you're someone who likes to make homemade foods, this is a great opportunity to maybe try batch cooking. And so you may pick one day a week to just cook a large portion of food and then portion it out and put it in maybe some freezer bags or some containers. And that way you have a bunch of meals ready to go and it saves you some more time on preparation and cooking. Now, home delivered meals and community meals are also another great option to ensure you're getting food available. Some of these might be require some different eligibility requirements. So it just might be good to talk with your ALS team, a social worker or case manager to see if you're eligible for any like home delivered meal services, such as like Meals on Wheels. Community meals may often be free, to the public. So there's, again, just another opportunity where you can get something hot or something fresh, especially if you're struggling financially, or again, just having just a more difficult time having some meals prepared or finding time in your day to prepare. Another important aspect is making sure you have appropriate equipment or adaptive equipment. So things like silverware, plates, mugs. This helps to make sure not only that if you not spill anything, but also promote independence with eating. Because again, as ALS progresses, it may just become more difficult to hold things, to making sure things are set up right. So work with your occupational therapist to see if there's any adaptive equipment that can be helpful to you. And then minimize distractions. It is really easily for us to maybe scroll on our phones or just maybe watch TV as we eat. But again, as ALS progresses and it just becomes much more of a task to eat, we want to make sure you're eating and drinking safely. And so focusing on maybe your meal at hand or your snack at hand will help mitigate any risks with that, but also to help you focus too on finishing what you have as best you can. And so I'll talk about next about using oral nutrition supplements. And so what I mean by this is that they're fortified nutrition products, and they help meet your nutrition needs. So these often come in a shake, a beverage, a powder, or a dessert. When you're looking for some nutrition supplements, you may look for the words plus or high calorie on the label, because again, these are going to give you some more calories within the same amount of volume. And again, we're really emphasizing eating as much calories as you can. Common supplements include Boost, Ensure, Carnation's Breakfast Essentials you may have heard of. And these often come in like a chocolate, vanilla, strawberry flavor, but there's also a lot more different types of flavors coming out. There are some plant-based options readily available as well. So Kate Farms, Complete, Orgain, Evolve are just a couple to name, but there are a lot on the market. If you're not someone who likes the thicker, creamier types of supplements, they do make some that are more like a fruit juice, so they're more of a clear liquid. Oral nutrition supplements are bought over the counter, so it doesn't require a prescription. They can easily be bought at a retail store, a grocery store, pharmacies, or even online through like Amazon or through the direct manufacturer's website if you're looking for something specific. So you may just have to look maybe in the health food section. You may even want to consider doing store brand supplements too, because these can be costly over time. If you are doing the store brand, supplements, that's great. Just make sure you're checking out the calories per serving, because again, we want to make sure you're meeting your calorie needs as best we can. Here I have a picture of a nutrition facts label. Most important thing to focus on with this label is going to be at the top. It's going to be that calories, the amount per serving, and then the serving size. Oftentimes, the serving size will just be one bottle or one container of what it is. So that's saying if you drink the whole bottle, you will have 240 calories. Sometimes the food label gets tricky. Sometimes they just say a serving size is half a bottle. And if that was the case, if this was half a bottle and the amount per serving of calories was still 240 calories, if you drank half the bottle, you would have 240 calories. And if you drink the whole bottle, you would have 480 calories. So you would double it. So that's the most important part of the food label to look for when you're looking for supplements. Don't worry if this is a little bit too confusing or you have more questions about it. That's what a dietitian is there for, is to help you navigate the different nutrition products and supplements available. Now, insurance may or may not cover part of the cost. It really just depends on the insurance plan and your coverage. Some insurances may consider this as another grocery food item, and so they may not see it as a medical necessity, whereas some insurances do. It doesn't hurt to look into whether your insurance covers or partly covers some of the cost. Work with your ALS team or and maybe a case manager or social worker to see if there's any potential in your insurance covering any of the supplements. And of course, talk with your dietitian to see which supplement is going to be best for you and how much to have each day, because again, there are so many different options out there. We want to make sure you're finding one that you enjoy having or you don't mind having, because you can get flavor fatigue after a while. But we just want to, again, making sure that you're meeting your nutritional needs as best you can. And so there are some nutrition supplements out there that are really high in protein, and they're great. They're great if you're not meeting your protein needs, but they just tend to be a little bit lower in calories as well. So just be aware of that if you're looking for nutrition supplements. But again, just touch base with your dietitian as well. Staying hydrated. So drink more fluids more often. Water is going to be a great source of fluids for you. But again, if you're able to sneak in any of those calorie-containing beverages, that's also wonderful. If you're someone who doesn't really like all those, heavier calorie beverages, and you like water, but you're getting tired of it, consider adding some citrus, fruit, splash of juice, flavor enhancers to your water, just to making sure you're drinking enough fluids throughout the day. Keep your fluids nearby and set reminders, whether it's like on a smartphone or a kitchen timer, because again, that's going to help prompt you to drink fluids. As well as having a good setup ready to go. You may want to consider having a couple water bottles easily available, or maybe even having a straw set up. So that way it just makes it a little bit more easier and more adaptable for you to drink throughout the day. And you may also want to consider choosing an electrolyte-containing drink, especially if you feel like you're drinking a lot of fluids overall, but you're just not feeling hydrated. Talk with your dietitian to see if there's any gaps in your day where you can maybe add more fluids, or even add like foods that are high in a water content, such as like cucumbers, melons, berries, citrus, etc. But if you're still struggling to stay hydrated, a dietitian can help you making sure you're staying hydrated with an electrolyte beverage. Now, staying hydrated, watch out for fluids that can dehydrate you. So think of anything that's really going to be caffeinated. So coffees, teas, some sodas that contain the caffeine, alcohol, and energy drinks. Those are the main culprits of fluids that are going to dehydrate you. Signs and symptoms to look for. So of course, feeling thirsty. Oftentimes when we feel thirsty, it's a little bit more of a delayed response in our body. So by the time we're feeling thirsty, our body has already been dehydrated. So making sure you drink throughout the day just to stay on top before you feel really, really thirsty. Dry mouth and dry lips are another sign. Dry or tempting skin. So in this picture here, In this example, you would just gently pinch the skin, like on your arm or your hand. It shouldn't really hurt a whole lot, but just gently pinch the skin together. And if you notice that your skin is kind of staying in that same position when you let go, that may be a sign that you're not getting enough fluids in. But if you're feeling really well hydrated, your skin should bounce back down to its original position. Now, dark yellow urine color or urinating less overall is another sign of dehydration, as well as constipation. And so with hydration, it's also just really important to, again, making sure you're staying on top of everything, as well as being aware of, when the weather gets a little bit warmer, or you're just sweating a little bit more, you're going to have more fluid needs. As we age, we also just tend to lose that sense of thirst a little bit faster. So again, just making sure you're staying on top before you exhibit those signs and symptoms. So constipation, like I said, it may be caused by dehydration, but it may also be just due to eating less fiber foods overall, just due to difficulty eating those textures. Less activity and exercise and having weaker abdominal muscles can also contribute to constipation. And although while ALS doesn't directly cause, issues with the GI tract, a lot of patients with ALS just exhibit that constipation. And there can be, again, multiple factors, including some certain medications as well. So how do you treat constipation? Again, making sure you stay hydrated. You may want to consider like a prune juice, a fruit and vegetable juice, incorporating fiber-rich foods into your diet. So fruits, vegetables, whole grains, beans, legumes, nuts, and seeds, if you can tolerate that texture. They're all going to be foods that give you fiber. And so if you're having a difficulty time chewing and swallowing some of those textures, consider choosing like a canned fruit or vegetable or cooking it down really well. Using a blender, food processor. For nuts and seeds, if you enjoy having that in your diet, but it's just something really difficult to eat, consider adding it to maybe yogurt or applesauce or something to help bind that food so that it's a little bit more safer to swallow. Fiber supplements may also be another option to treat constipation as well as bowel medications, but we would recommend talking with your doctor first about the various options and which one is best for you. Now, what happens when chewing and swallowing get worse? This is also known as dysphagia, and it may increase that risk of aspirating or just choking or coughing when food goes down the wrong way, because again, with ALS progressing, it just affects those muscles involved with chewing and swallowing. So we recommend, if you're noticing that it's getting more difficult, keep foods really small, keep them really soft, and add moisture wherever you can. So again, adding broths, gravies, extra butters or fats to foods just to help keep things moist and soft and smooth going down when you swallow. You may want to consider having a food blender or a food processor available, either to pulse foods into smaller chunks or even a puree texture if that's a little bit more easier for you. You may need to avoid more dry, crumbly foods or just more thinner, runny liquids. You may need to use a thickener for liquids, but would recommend just working with your SLP on further food or liquid diet modifications because they're going to be the experts in what textures are most safe and appropriate for you. As dysphagia continues to progress, it may be a point where you just want to continue to eat the foods you enjoy the most, and that's totally okay. It might also be the time when you might want to start considering about whether a feeding tube is within your goals of care or not. And I'll kind of explore that a little bit more in detail. So when should a feeding tube be considered? As ALS starts to affect the muscles involved with eating, so chewing, swallowing, and also moving or manipulating the food in your mouth. When you're unable to meet your nutritional needs, when hydration is poor, when weight loss and malnutrition is present or of concern, when eating becomes fatigue, tiresome, and burdensome, and before your respiratory function is too poor. Because a feeding tube is going to require a procedure, we want to make sure that your body and your respiratory function are okay first, before deciding it might just be a little bit too late to get a feeding tube. So it's best to have these discussions early on as you decide whether a feeding tube is within your goals of care or not. So why should I get a feeding tube? It does help meet 100% of your calorie, your protein, and your fluid needs. So it can help lift some of that pressure or some of that burdensome to meet your needs by mouth. If you're eating less by mouth, maybe choosing not to eat by mouth at all, or just only selecting the foods you enjoy, it can help decrease your risk of aspirating from oral intake. It's easier to take some medications through the tube. Check with your doctor first, just to make sure if there's another form available for that medication, or if it's appropriate to flush through the tube. Some medications may be too big and might clog the tube, so just check with your doctor first. Having a feeding tube can save you energy, and again, just allowing you just to enjoy the foods you like to taste. But that way you can spend less time eating and more time doing the things that you love, or spending more time with loved ones. So what to know about a feeding tube? Again, it does require a procedure to insert a small flexible plastic tube through the abdomen. We often call this a PEG or a G tube, and it helps to deliver the formula, hydration, often as water, and some medications that are appropriate to be flushed through the tube. So in this picture here, I have an example of a feeding tube. And so you can see that there is a little internal balloon or a bumper that would be on the inside of your body, and that just prevents the tube from falling out. And then we have another bumper on the outside of the body, and that's to prevent the tube from falling too far in. So that helps to anchor the tube in place. It does extend a little bit out of the body, but that way it helps for you to, connect the parts and pieces. But this tube can be securely fastened around the abdomen or around your belly. It can be done so with some special tape. It can be done with like a mesh netting, so that way you can kind of tuck your tube in, so it's not flailing around or getting caught on anything like as you sleep. So there are ways to secure it. And those are the most common standard type of feeding tubes. This picture is a low profile feeding tube, or I like to call it a button tube. And it's just a lot shorter. There's no big extension outside of the body. So the different delivery methods to making sure you're getting nutrition through the tube would be through a syringe, a gravity bag, or a feeding pump. And so what this looks like, syringe, again, you can draw up the formula, drop the water, you would connect it to your feeding tube, and then you would just push it in, and any medications would need to be flushed with a syringe. You can also choose a gravity bag if you want to just have a slower rate of being fed. So again, formula can go in, hydration or water can go in, but we do not recommend medications to go in. We only like to have those medications be able to be flushed with a syringe, because we don't want the medication to clog in the tube lining. These are going to be the two common methods to give feedings and give hydration for patients with a PEG or a G tube. And people may be nervous about, syringing through their loved one's tube, or just connecting them to a bag and letting gravity do the work. You would just set the bag above the head, whether it's an IV pole or like a coat hanger hung on something to help let things drip through with a rolling dial. But really, when we think about eating a meal, let's say we have a protein at dinner, like a chicken, a starch such as potatoes, and maybe some green beans on the side, and maybe a glass of milk. That's going to be at least like 2 cups or more of volume. And so we often maybe eat in 20, 30 minutes, or maybe a little bit longer. And usually when we're giving tube feedings, we may only give 1 or 2 cups of formula at a time. So it's not too much different in terms of volume as it would be with eating by mouth. Now, if for some reason you're not able to tolerate those 2 methods of feeding, if it just feels like you're really bloated, it just feels uncomfortable, or it just goes really too fast, there is the option of a feeding pump. So it's very similar to the gravity bag method. where you'd pour the formula and water into the bag. And then instead of using like a little rolling dial to control the speed, it would be set up with an electronic pump. And that way you can precisely control the rate of how fast or slow you're feeding into the tube. Now this example shows 60, this would refer to 60 milliliters per hour. And so that's about a quarter cup. And so if we think about a quarter cup with like a measuring utensil in your kitchen, think about that volume and then slowly dripping that into the stomach over a whole hour. So it's going pretty slow. It's only going to have a couple drops, every few seconds or a few minutes over the course of an hour. So it really just helps to slow it down. It's another option for those who just don't tolerate the first two methods, or if you have an altered GI tract or another medical reason, to have a feeding pump, it is an option as well. Similar to the gravity bag, we would not put medications through the bag or with a feeding pump. All medications would need to go through with a syringe. Frequently asked questions about a feeding tube. Can I still eat and drink with a tube? Yes, absolutely. Like I mentioned before, you just may want to choose eating the foods that you like to taste, and again, saves you some energy and offloads some of that burden and pressure from meeting your nutrition needs by mouth. Can I stop using the feeding tube at any time? Yes. If you feel like that it's just getting to be too much of a hassle, you just no longer want to depend on tube feeds, that's okay, and you can choose to just not use it. Can the feeding tube be removed if I don't want it anymore? Yes. It may depend on the way the feeding tube was placed, but yes, it can be removed. It doesn't have to be permanent. Can others see that I have this tube? No. It can be securely fastened around your waist, like I mentioned before, and then hidden underneath your shirt. So it's really not obvious that you have a feeding tube. And oftentimes you would just have that feeding, at home, where you're able to connect with a syringe or with a feeding bag as well. What risks are there to getting a feeding tube, and can it fall out? There's going to be risks, as with any procedure. So yes, there is going to be a possible risk for infection or just some pain or redness or irritation at the site where the tube is, or maybe the tube gets clogged. These all can be treated, but just like with any medication or with any surgical procedure, there are always going to be risks, and it's going to be something to, you may want to weigh as you're making that decision. But best to have those discussions as well, those risks with your doctor and your ALS team. In terms of the tube falling out, it is really uncommon for those PEG or G tubes to fall because of those little anchoring devices inside and outside of the body. But if the tube does fall out, it can be replaced. We would just recommend for you to have that feeding tube that fell out, place it back into the hole or the site where it was, and that way it keeps the tunnel or the track into your body open. So think of it like an earring. If we removed an earring and we just never had anything through the hole, it would eventually close up. And we don't want that to happen with a site going into the stomach, because we want to be able to replace the feeding tube. So if it does fall out, put it into the site, but just don't use it. So don't do any water flushes, don't do any formula, don't even put any medications through, because we don't know where that tube is going to end. We would want you to go to the nearest urgent care or emergency room, so that way you can get that tube replaced and then recheck the positioning of it to make sure it's safe to be able to use again. But again, we just want to keep that site, that track open. So feeding tubes can be replaced, but it is very uncommon. How do I clean the feeding tube? Pretty easy. Just use that syringe and just flush with clean water frequently. So you would flush before and after you would do a feeding, before and after every medication, or maybe a couple times throughout the day. Not only does it help give you some more hydration, but it just helps to keep the tube clean and clear. You may be able to use a little small brush around the end of the tube. It just helps to keep any of the debris out or any crusting or dried formula clear. You can use that brush and just soak it into mouthwash just to keep it sanitized, or even just toss and get a new one. It often comes in the supplies with your tube feeding needs. How do I clean the site or the skin around the feeding tube? You would want to close the port or the end cap of your tube so that nothing gets in it. But you would just shower and wash up like you normally would. So warm water, mild soap, or even a washcloth around the site is fine. Keeping that site open to air is going to be best for the skin. Might be a little irritated right after you get a feeding tube placed, but a couple of days after it should be okay. If there is continuing irritation, redness, or leakage from the tube site, talk with your doctor or your nurse to have it evaluated and to see if there's any treatments that should be done. What does the procedure look like for getting a PEG tube or a G tube? So like I mentioned, it is a procedure, so it does require some mild sedation or being under anesthesia. It's typically followed by an overnight observation at your local hospital. And there you'll receive some education by a nurse and a dietitian on what your nutrition regimen is, how to use the tube, and how to use the supplies that go with it, and then proper site care in more detail before you discharge. Now, where do I get those feeding tube supplies, like the syringes and the formula, etc.? So a dietitian will help calculate how much and what kind of formula you need to help making sure you're meeting all your nutrition needs. Your hospital team usually helps with the initial setup to arrange supplies for going home just for a couple of days. But your formula and supply coverage is going to be run through your insurance, so the costs will vary. But you'll be set up with a durable medical equipment company or a DME, and they'll help ship and deliver all that formula, syringes, bags, cleaning pieces, connecting tubes, anything that you need straight to your home. So if you're starting to run low on supplies or formula, you would just give your DME company a call and just let them know, and that way they'll continue to ship all your supplies that you need straight to your home at your doorstep. Now, after all of this, should you still get a feeding tube? And that's really going to be up to you. There is no right or wrong decision about a feeding tube because everyone is going to have individual reasons why or why not to get a feeding tube. And we just want to let you know that this is an option to help meet your nutrition needs, but you don't have to because you can make all the decisions about your care. We just want to let you know of what that might look like. Again, having those discussions with your doctor, what are the risks versus the benefits? So again, have those discussions early or maybe just start thinking about, do I want a feeding tube down the road? Maybe sooner than you thought, because again, we just want to make sure if you do want to have a feeding tube that you're able to get it safely. Next, I'll move on, vitamins and minerals for ALS. Now, this is a really popular topic in research. There is so much mixed evidence for specific nutrients that slow the progression of ALS. A lot of research is leaning towards certain vitamins and minerals, but a lot of them theorize, if it can slow the progression, or are there certain nutrients or things in the diet that may, cause ALS or may prevent it. But again, the evidence is just so mixed. There really is not a conclusive nutrient that we would recommend specifically at this time. So we would recommend, though, eating a balanced diet just to make sure you get all those food groups in as best you can to help all of your vitamin and mineral needs be met. So including fruits, grains, starches, vegetables, protein foods, dairy, and healthy fats. You may need a multivitamin with minerals or maybe even individual supplementation if you're deficient for a micronutrient or if there are gaps in your diet, but your dietitian and your doctor with your ALS team are going to be the best ones to just talk about supplementation first before you take it. Or if you do have any further questions on vitamin and mineral supplementation, again, let your dietitian know because they can help evaluate your diet and see, do you need the supplementation or not? Or if so, what's an appropriate dose to have? Some other resources I included aside from the MDA and the ALS Association, the Academy of Nutrition and Dietetics. Now, they don't really have specific nutrition recommendations for ALS, but they do offer some really good resources and recipes on making sure you have that balanced diet, or just some ideas of different foods to include in your diet, or even like specific nutrients if you feel like you're not getting enough of certain ones, or if your dietitian tells you, we need to make sure you're getting enough, vitamin B, things like that. So more of a just general overall good resource on making sure you have a balanced diet. The OLI Foundation is another great resource, specifically for those with tube feedings. They do a lot of great work in, again, talking about, those commonly asked questions, troubleshooting some problems, support groups, and other resources for those with tube feeding needs. And then you may have heard of ALS Untangled. Now this is run by the North American ALS Research Group, and it's made up of a bunch of clinicians and scientists, and they do a lot of the heavy lifting or work, looking into the research and looking into the literature of a lot of alternative or off-label treatments or supplementations that people are looking at specifically for ALS. Now they do their own protocols of reviewing and ranking different treatment options, and they also take into consideration too reviews from those with ALS and what the outcomes were. And they kind of sift through all that and kind of list out a little bit more clearly what does maybe the research show, what are some reported symptoms or outcomes. And this is more just to provide some information if you were interested or looking into those more alternative off label treatments or supplements. Not so much to tell you yes or no to take this, but really just what does the current research and literature say. And if you are interested in taking any of those treatments or supplements, we highly recommend just talking with your ALS team and your doctor about those before you take it, just because sometimes those supplements and treatments may interact with medications, and we just want to make sure that we take a look at any of those before you start taking them. So I know this is a lot of information, but if there's anything you can take away from tonight, even if it's one thing, that is my goal and hope for this presentation. So some things I really like to highlight, again, is a dietitian can help those with ALS and their caregivers navigate their nutrition care. Good nutrition can help improve the quality of life for those with ALS. Eat more calories, don't shy away. Stay hydrated. Maintain your weight as best you can. Oral nutrition supplements and feeding tubes are optional and completely up to you to use or not. And choose and enjoy eating the foods you love the most. So again, if there's anything you can take away, even if it's one thing tonight, that would be great. Or if you found something else interesting that you learned tonight, that is, again, my goal and hope for this presentation. These are my references that I used for this presentation, but I would just like to thank you all for watching and thank the MDA for hosting this webinar. But otherwise, I'll open it up to questions.
Thank you so much, Sarah. That was a really informative presentation on nutrition and how we can navigate that when dealing with ALS. I do think we have a few questions for you here. Let me just share my screen again. And then, so the first question I have for you is, how can someone ensure that they're getting enough hydration? You talked a lot about hydration and the importance of staying hydrated, how to check to make sure that you're hydrated. But as swallowing becomes more of an issue, how can someone ensure that they're staying hydrated to the best of their abilities?
Yeah, so again, we do have some methods of estimating how much fluid needs you would need. And again, we would take that into consideration with our nutrition assessment, but really it's just going to be kind of like those signs and symptoms to tell your body telling you, am I feeling hydrated or not? And based on, individual report, if you're having any of those signs and symptoms, as that chewing and swallowing become more difficult, it is really going to be hard to meet your hydration needs. Even with thickened liquids, if it helps with the swallowing, oftentimes thickened liquids, people really fill up pretty fast on it, and people do get still dehydrated or still a little bit behind on their hydration, even with a modified liquid texture. But if you can add any liquids to foods, like broths for more savory items, or more water or juices to anything. And again, just modifying that texture as best you can where you can tolerate it will help meet it. But, you know, if dysphagia continues to worsen and you're just still chronically dehydrated, again, it just might be a point where you might want to consider having a feeding tube. But if not, you know, just do the best you can, you know, drinking as much as you're able to.
Great, thank you. So what are some signs that may indicate that someone needs to adjust their diet or seek further assistance?
Sure. And is this going to be more so for like, you know, specific nutrients and like food groups, or just overall, like, do I need to make adjustments for?
Yeah, just if someone is feeling like wrong in their body or is starting to see decline happen at a faster rate, and they're kind of seeing that, what kind of adjustments could they make to their diet, or when should they know to seek further assistance?
Yeah. So weight is a good indicator. It's not the best indicator, but weight can often tell us, are we meeting our nutrition needs, really calorie needs, to make sure, you know, we're getting enough in. When we ask, you know, for like a diet history or a diet recall, again, we're kind of looking at, are you able to get different food groups into your diet? If not, what are some foods that you might be interested in having to making sure you hit those needs? So if you don't like vegetables, are there any vegetables that you may like once in a while, or maybe we can add, you know, to your diet that doesn't taste as off-putting or as strong? Again, just to making sure you get that balanced diet. We really work with you in what you typically eat in a day and what can we add already. Not so much of let's erase everything, start over, you know, start eating differently. It's more so about what else can we add to what you like to enjoy to eat, what you have available, because we really want to make it more individualized, you know, for each patient and individual. So we always like to focus on, you know, can we sneak in an extra snack? Can we add like maybe an extra tablespoon of butter or oil somewhere into your diet? You know, if you're at ALS clinic, do they have like a scale appropriate even for those with wheelchairs? So that way we can continually assess, you know, their calorie needs or their weight trends and making sure they're meeting their calorie needs. Having a diet recall, sometimes we might do kind of like a calorie count. So we might ask a little bit more specifically about portions, about cooking methods, you know, other things that you add into meal preparation, and we can kind of calculate a rough estimate to calculate what you're meeting. It's another tool. It's not going to be perfect. No method is ever going to be perfect. Otherwise, you know, we would be using it all the time. But it's just another way. Can we compare what your diet recall says to what we're estimating your needs to be? And sometimes they are off, and sometimes, you know, patients may be underreporting or overreporting what they actually eat. So a lot of it is just going to be, you know, asking questions from different angles to really help gauge what else can we do to making sure you're meeting your nutrition. In terms of when to seek help, you know, even if you feel like you're eating really well, you know, you're maintaining your weight, still seeing a dietitian early on doesn't hurt. Again, sometimes it's nice to have those little reminders, you know, also just to discuss, you know, some options, just to think about, you know, maybe before you start losing that weight, because it is really hard to play catch up with nutrition, especially when people are losing weight. So even just talking about like, hey, have you ever considered maybe trying like an extra nutrition shake? Maybe just to kind of experiment, you know, what's out there, what flavors you like, something else to maybe add in slowly to your diet. So I think seeking a dietitian earlier is going to be better than later.
Great. And that kind of leads into another question that I have for you, which is if someone doesn't already have a dietitian on their team, what are some avenues to access a dietitian? And are there telehealth opportunities available or in clinic? And if you can talk to that a little bit.
Yeah, so I would strongly recommend talking maybe with your PCP or your primary care provider as well, because they can also help send a referral to a local dietitian in your area. And help you set up, you know, seeking a dietitian. telehealth can be an option. It does get a little tricky just with different state licensures for dietitians, but it doesn't mean that dietitians can't be licensed in multiple states. But I think going through your PCP to get you set up with a dietitian is going to be best. Seeing a dietitian like in an outpatient setting, again, costs are going to just vary depending on your insurance, what they take for insurance. So sometimes you might be able to get in to see a dietitian without maybe a referral from your PCP, but I think that would be my first step, just to see, who can I connect with locally.
Great, thank you. And just one last question, and I'm not sure if you know the answer to this, that's okay, but just thought I would ask. Do you know of any ongoing research or clinical trials that explore diet and nutrition in ALS management? And are there any that our community should be aware of, and how to access those?
Yeah. So I will say I'm pretty, new diving into the whole ALS and, nutrition research of it. I can't say any off the top of my head, but that's not to say that there aren't any ongoing right now. So definitely something I will need to look into myself. But I know sometimes I've seen some research where they look at maybe like different types of tube feeding formulas, or just looking at more of a retrospective study of different diets. But I don't know of any directly off the top of my head for clinical trials. So definitely something to look into myself.
No, that's great information. Thank you. And I think we have Dr. Ostrow in the chat, kind of giving us a little heads up as well. So he's still thinking about that. And so those of us who are still on the call, if you want to look in the chat, Dr. Ostrow is giving us a little information there too. So I don't think we have any other questions for you, Sarah. So thank you so much for your presentation today. It was very informative, and you got a lot of accolades in our chat. We sincerely appreciate your time and your expertise and your support of the neuromuscular community. Thank you, Sarah.
Thank you.
If you are new to MDA and have been diagnosed with a neuromuscular disease under MDA's umbrella, or if you or a loved one of someone who is diagnosed, we encourage you to stay engaged with us at MDA. You can do this by visiting mda.org/join and completing a short form. Joining is free and will keep you up to date on the latest research, educational programs, and supports. MDA partners with the Center for Disease Control and Prevention to promote the National ALS Registry, the only national population-based registry in the U.S. that collects information to help scientists learn more about who gets ALS and what may cause it. The National ALS Registry is a program to collect and analyze data about persons living with ALS. Signing up is free and only takes a few minutes. We have several programs coming up that you might be of interest, including next week's Advocacy Institute with MDA's Advocacy and Public Policy team that we talked with Paul about earlier. They will be sharing updates from the Hill on ALS policies. Then later on in the year, we will be offering a next step seminar for people who have been recently diagnosed with ALS. This virtual seminar aims to provide information that is manageable and actionable while helping families to connect with others in the community. Lastly, we are offering an in-person symposium in several cities this year. Please scan the QR codes on the screen or click the links in the chat to learn more about this. Again, we would like to thank Mitsubishi Tanabe Pharma America for supporting today's webinar. We would love to hear your comments about today's session of our webinar series. Once you leave the webinar, a web page will automatically pop up with a short survey about today's webinar. Completing the survey will provide us with important insight as we plan for future educational programming. So please fill that survey out as you're leaving. If anyone has questions after this webinar, please feel free to contact MDA's Resource Center at 1-833-ASK-MDA1 or email our Resource Center at resourcecenter@mdausa.org, and we will be sure to get back to you. Thank you again to Dr. Ostrow, Paul Melmeyer, and Sarah Dramstad for your time, and thank you to everyone who attended day 1 of our ALS Learning Series. This concludes today's webinar. We hope to have you back here tomorrow for the second day of our ALS Virtual Learning Series. Have a wonderful evening.